بسم الله الرحمن الرحيم. بنبدأ اليوم أولى فقرات مؤتمر كلية الطب بعنوان Mansoura University Regenerative Medicine Program from Bench to Bedside. برحب ترحيب بالسادة الحاضرين، بالسادة العملاء والسادة الوكلاء وجميع ضيوف المؤتمر وجميع الباحثين وجميع القائمين على إعداد هذا المؤتمر. نبدأ أول سيشن في المؤتمر بيرقصوا الchairpersons لهذه first session البروفيسور ماجدة نصر from Faculty of Pharmacy, Professor Azza Ismail from Faculty of Science, Professor Muhammad Yusuf from Faculty of Veterinary Medicine. أولى هذه السيشن بتتضمن 5 فقرات، أولها بعنوان Cancer Stem Cells اللي هيكون بالإلقاء من البروفيسور حسن عبد الغفار، فليبدأ بإلقاء البحث الخاص به. يا ريت الأفيلبل تايم بالنسبة لحضرتك يكون في حدود من ربع ساعة لثلث ساعة أظن. نلتزم بالوقت إن شاء الله، وبعد كده نبدأ الدسكشنز في 5 minutes.
Cancer stem cell acute treatment, and the other one is cancer achieving cells in breast cancer in collaboration with the medical community center. As you all know, the stem cell is a primitive and differentiated cell that gives rise to other types of cells, and also has very important or very unique 3 characteristics. The first one is the internal self-renewal. The second one is plasticity, it means trans-differentiation from any lineage to another, or even from the hematopoietic one can trans-differentiate is the hepatocyte, and the neural cell can trans-differentiate is the hematological, which is recently known. The third one is a homing. I mean by the homing, for example, if there is cardiac ischemia and we take stem cell in the peripheral blood, we find, for example, after 2 hours, this injected cells in the cardiac muscle. This is a very known hierarchy for hematopoietic cells. As you all know, this hierarchy, the recent one known is about the stromal cell microenvironment, which has now, as we hear today and tomorrow, many talks about the role of this mesenchymal stem cell in the future of medicine. One of the very important issues is when we are using the stem cells, it's very important to use a pluripotential line, because this pluripotential line has the unique characteristic of the stem cell, other than the progenitors or the more mature one. So before injection of cells, if we are planning to inject a pluripotential stem cell, we have to have these cells with this characterization. For example, these cells should be in T0 phase, 5 neural cell resistance, colony forming unit stream on day 16 to 20. All of these characteristics should be fulfilled before we say this is a pluripotential stem cell. Otherwise, the injected cells, we need to re-inject the cells maybe after 6 months again, maybe after 1 year, I don't know. We are shifting now about this carcinogenesis. As we all know, the initiation of cancer has many theories. The first one is the failure of genetic mechanism that controls the growth and proliferation of cells. The other one is the cumulative damage to multiple genes, we call it multiple hit model. The other one is about the aggressiveness and secondary tumors, and all of these depend on the cancer initiating cell. The problem of metastasis, the problem of relapse in malignancy, this is because the chemotherapy or the protocol used for management not indicating the primary cell which is responsible about this tumor. So the future target therapy for cancer is aiming to eradicate these cells according to the type of the tumor. For example, in the leukoplastia, there is now known cells with simple marker could be isolated and using for target therapy and in other in vitro and ex vivo to apply in future on the human trials. This model also, this cancer stem cell, a minority of cancer stem cell with tumorogenic potential. This apparent regulation in the self-renewal or differentiation of regenerative ability. What are the cancer stem cells? The cancer stem cell is mimic the normal stem cell in many criteria. The self-renewal, the normal stem cell has self-renewal capacity. Also, cancer stem cell has the same ability. Differentiation into multiple cell types. Also, you can apply on normal and cancer stem cell. The third one is from a distinctive population in humans that likely causes the disease relapse and metastasis. So understanding the self-renewal of cancer stem cell is very important for future target therapy. Also other properties, sharing of normal stem cell and cancer stem cell, like as we said, self-renewal, differentiation into phenotypical diverse mature cell types, and also the regulation. This is very important. The regulation of both normal stem cell and the cancer stem cell are the same. I mean, perturbation or abnormality in this regulation is the main cause for to develop cancer initiating cells. We are going to now to have the cancer stem cell models. There is two main models for cancer stem cell. Either stochastic model or cancer stem cell model. As we know, this stochastic model, any cells in the tumor can develop appreciate tumor. But I think the second model, which is the more accepted model now, which is a cancer stem cell model. I mean, within the tumor, only certain cells have the ability to initiate this tumor, and it is responsible about the relapse if the chemotherapy or the target therapy doesn't affect this cell, as we see in the cancer stem cell model. And in this cancer stem cell model, every type of tumor has its own characteristic cancer initiating cell. For example, for the breast cancer, there is certain, now we can isolate these cells and put it in vitro or in vivo for target therapy. For example, there is a work now running for this cancer, for example, breast cancer stem cell, which is CD44 positive and CD24 negative. So these cells, when we isolate this target and inject in the nude mice, it regenerates and initiates the breast cancer. Molecular pathway of cancer stem cells. The molecular pathway for stem cell differentiation are complex. Indicated that this regulation could occur at multiple sites to turn off the hemostatic balance and create abnormal cell or cancer cell. These are the more important 5 genes about which is controlling the cancer, the normal stem cell pathway, and its perturbation cause the cancer stem cell. And even now, there is now clinical application for these target in target therapy of cancer. For example, this is the main 5 genes is the main gene. The second one is the H4 gene. The third one is EMI gene. The third one is Munch gene. The fifth one is Baton gene. This is five important genes controlling the proliferation and the control of normal stem cell, and its perturbation causes malignancy. And not, and if one, for example, is concept about certain type of, for example, now anti-notch now used for treatment of chronic leukemia. As we see here in this diagram, pathways involved in self-renewal that are very curated in cancer cells. For example, the first one is the Wnt gene. This perturbation is the Wnt gene causes colonic carcinoma and epidermal tumors. For example, the Hedgehog gene causes retinoblastoma and breast cancer cell carcinoma. The third one is the Notch gene is responsible about the initiation of leukemia and also mammary tumors. Still having the molecular pathway in normal and cancer stem cells, and as we said, the Wnt, the Notch, the Hedgehog, and all of these act, as we see in this diagram, on the receptors, activate the responsible promoter according to the type of the tumor working on. The most important thing is to know all of the five genes not separated from each other. They are interacting with each other. For example, as you see in this diagram, the Hedgehog signaling network, there is a communication forward and backward between it and the Notch signaling pathway, and also through the Wnt pathway, and there is interconnection between the Notch and the Wnt, and all of these act on the stem cell self-renewal, and also the Hedgehog act on the BMR gene. This is diagram illustrates the previous talk about all of these genes, and all of these genes now having in certain types of tumor, it's a target and antagonist to treat certain tumors. For example, as I said, for the ABC transporter, drug resistance genes are using now for a target therapy in certain type of leukemia, and the same also for Notch pathway, for example, certain type of leukemia, and Wnt pathway and Hedgehog pathway. Finally, the cancer stem cell as invade, self-renew, and differentiate within tumors to form additional cancer stem cell, as well as non-tumorigenic cancer stem cell, which in oral, which have limited proliferative potential. As a tumor grows, this cell can either undergo limited benign growth or form disseminated malignancy. Identification and the characterization of cancer stem cell should therefore also lead to diagnostic methods that can distinguish between disseminated tumorigenic and non-tumorigenic cells. And finally, the stem cell specific and conventional cancer cell. As we see, the standard cancer therapy is like mowing the weed. It gets rid of the disease transient, but the cell always grow back. We need to get rid of the root to cure the disease, and the root here is the cancer-initiating cell. If we are eradicating the cancer-initiating cell, for example, in colon cancer or breast cancer or whatever, this is we have the standard the assurance that this will not relapse again. Thank you.
بنشكر حضرتك دكتور حسن عبد الغفار على التزامك بالوقت، ودلوقتي بنفتح مجال المناقشه الفلور. وبليز يعني anyone first introduce yourself to the world. كل واحد يعرف نفسه الأول قبل أي أسئلة. في حد عايز يبتدي أسئلة؟ اتفضل يا فندم.
Morning everybody, thank you Dr. Hassan. Very simply, malignant cells are nothing more than a deep differentiation. So the cells in the mature status range from a stem cell and then differentiate along different pathways to become a mature cell. What happens in most malignancies is that it goes back away. Okay? So your main focus is there is a way to go again from being differentiated into de-differentiated, and from being de-differentiated into differentiated again. This is the message of malignancy, and thank you very much.
There is in certain, as you said, in certain, not all the cancer-initiating cells come from the precursors, I mean from the stem cell to the brain. It could be from the mature cell. Like some studies now on acute leukemia, so the studies, when you study that, you don't only depend on the cancer, the precursor cell. In acute leukemia, the cancer is from the mature cell. And also there is, as you know, there is work now going on to differentiate from the mature cell to the stem cell to overcome the embryonic stem cell. And already I know that in the States working on this for thalassemia patient. They have its own mature cell and differentiate the cell to stem cell to create thalassemia.
Thank you.
May I ask a question if there is a difference in definition between cancer stem cells and tumor initiating cells and also site population cells? What is the difference between the three technologies? Cancer stem cells, difference between different immunologies. Cancer stem cells and the tumor initiating cells and the site population cells. What is the difference between the three immunologies?
The cancer initiating cell I think it is different nomenclature, but I think the same.
I think there is a difference between tumor initiating cells and the cancer stem cells.
Which form?
Cancer stem cells are stem cells acquire cancer productivity, or a cancer cell acquire a stem cell productivity. But I think tumor initiating cells are cells that when isolated from certain tumor has the power to generate another tumor with a small number. For example, if we have
another type of tumor, you mean?
No, another tumor but with a small number. If I have a tumor and I isolated these tumor initiating cells in a different model, for example, P53 non-mice, these cells has the power to give rise to hepatocellular carcinoma. So we have cancer cells and we have tumor initiating cells.
To make it clear, now cancer stem cell there is many studies that we should shift to not cancer stem cell to go to what we call pre-cancer stem cell. I mean, there is more studies now verify that the cancer stem cell it is not as this it is in the stage we have to look for the cancer in the pre-cancer status, not in the when there is ability to form. But I think for your definition about this tumor itself, it depends on because in some tumors and some malignancy now, not only one type of cell can initiate this malignancy. That's why maybe it is different. But I think when we confer that in this type of malignancy only one cell or one type of cell can initiate, I think it will go back to this cancer initiation.
Thank you.
The second paper is called embryonic stem cells. Neural stem cells. Professor Dr. Hani Mari, Professor of Cytology.
First of all, I would like to thank Professor Dr. Kagahan, Vice President from Mansoura University, Professor Dr. Mohammed Hussein, the Dean of our faculty, Professor Dr. Azza screen, and I'd like to express my deep appreciation to Dr. Mohammed for his kind invitation to participate in this meeting. My lecture, my presentation will be divided into 4 sections, 4 parts. The first part, I will talk briefly about the different parts of the brain, controlling the basic life processes. So as you know, the human brain is subdivided into 4 regions, and these regions control every part, every process. So if we look at our human brain is subdivided here into central core region, limbic system, and the cerebral cortex. This region controls different life processes. For example, if we talk about the limbic system, very important, this area controls the basic life processes like breathing, like pulse, and so on. Actually, I'd like to concentrate on the limbic system and concentrate on the hippocampus, which is the region especially affected in Alzheimer's disease. And my topic is about neural stem cells. So, neural stem cells, and as you know, Alzheimer's disease occurs due to degenerations of cholinergic neurons in the brain. Briefly, I will talk about the basics of a stem cell. Stem cells are cells which have the power to differentiate, self-renew, and differentiate, giving rise to other kinds of cells. This is an undifferentiated cell. These are the 3 main criteria characteristic any stem cells. And we have 2 types of stem cells, pluripotent stem cell and the multipotent stem cell. And the difference between 2 types is concerning the degree of differentiation. So, pluripotent stem cell has the power to differentiate, giving rise to different kinds of cells, while the multipotent cells have restricted degree of differentiation. So we have pluripotent and the multipotent. Let's now move to the neural stem cell. What are the neural stem cells? As you see, neural stem cells, we born with a fixed number of neurons in our brain, and every cells in the brain die, but the neuron have no ability to regenerate itself. That is why we need to look for cells which have the power to regenerate the defective area. In our project, we have selected olfactory bulb, adult olfactory bulb human neural stem cell, and these cells have the ability to self-renew, have the ability to differentiate, giving rise to 3 different types of brain cells, which are neurons, oligodendrocytes, and astrocytes. Why olfactory bulb? Number 1, easy access. Number 2, the cells are undifferentiated. This means that they have the ability to differentiate. From where we can get a neural stem cell, first source is olfactory bulb. We can get neural stem cell through an open protocol called direct reprogramming. One transcription factor called SOX has succeeded to transform somatic cell like fibroblasts into induced neural stem cells. And as you see here, these cells have the power to give rise to the other kind of cells. So use neural stem cell, very novel protocol by which we can transform somatic cells into neural stem cells. As I have mentioned, olfactory bulb and other stem cell in general, we use it to avoid the ethical issue associated with the collections of embryonic neural stem cell from embryos. In our project, we have isolated human olfactory neural stem cell from the olfactory bulb, we have characterized these cells as immunohistochemical and gene expression profile, and they have made 3 models, 3 animal models. The first model is the Parkinson model by using 6-hydroxydopamine, and the second model we have used ibitonic acid to develop an Alzheimer model, rat Alzheimer model, and third model was the spinal cord model by destructions of certain region of the spinal cord, and we have injected our cells in the 3 models, and the good news is that our cells have succeeded to restore the functions. In case of Alzheimer model, we have noticed a significant difference in the improvement. If we look at the 6-hydroxydopamine by injections of our cells, and our cell was genetically engineered to produce nerve growth factor and enhanced green fluorescent protein. The end result of this experiment, we have done behavioral assessment using rotor test and ball test. These are the tests here, and we have noticed a significant difference and significant improvement in the sensory motor functions between the treated and the non-treated group. Also, in case of our Alzheimer model, as I mentioned, we have used ibitonic acid to destroy the cholinergic neuron within the hippocampal region, and the engraftment of our cell in the hippocampus of the ibitonic acid treated rat, there was a significant difference. We have made a behavior assessment using Morris water maze, and we have noticed a significant improvement in the cognitive and the memory functions between the treated and the non-treated group. Unfortunately for the spinal cord injury, we have also injected our neural stem cells, which are genetically programmed to produce nerve growth factor and enhance green fluorescent protein. Unfortunately, there was no significant difference between the treated and the non-treated group. And our explanation for this, that as we know, we have isolated the neural stem cell from the brain. And the niche in the brain is different from spinal cord. That's why our cells, which are separated from and isolated from the brain, gave a positive result when we grafted them in the hippocampus and striatum. Now we are working on immunohistochemical assessment of the 3 experiments. We are going to use about more than 20 markers to test the differentiation, the survivability, and the ability to restore function after engraftment. Briefly, this is our lab at Faculty of Veterinary Medicine. This is the process of isolation of olfactory bulb neural stem cell. We have collected the brain tissue from Italy, Catholic University, Italy, and we have succeeded to isolate the neural stem cell, and we have shipped the neural stem cell from Italy to Egypt, where we continue our experiment. This is the neural stem cell lab at Faculty of Veterinary Medicine. This is a transfector, and this is a liquid nitrogen storage. This is a process of incubation, and this is a neurosphere. These are the neurosphere which are formed after isolation of neural stem cell. These are neurosphere for human olfactory bulb neural stem cells. Second, we have made immunohistochemical characterization of these cells. As you see here, this is the process of differentiation. The cells here are undifferentiated. They are positive for nestin. These cells are positive for NG2, which is marker for immature oligodendrocyte. And the next slide, we will see here positive also for NG2, which is immature marker for oligodendrocyte. The next slide, we will continue the process of differentiation of neural stem cell. As we see here, positive reactivity, green color for NG2, and I think in the next slide we will see the process of neuronal differentiation. The neuron here, the red color, this is the positive reaction for microtubule associated protein, the red color one, and the blue color ones are differentiated astrocyte. And as we see here, the percent of neuron are very few in comparison to glial cells and astrocytes. Here also, positive reaction for microtubule associated protein and glial fibrillary acidic protein. So this is the immunohistochemical analysis of our human olfactory bulb neural stem cell, and from this analysis, very clear that neural stem cells are multipotent. This means that they have the ability to differentiate, giving rise to the main cell type which forms the nervous tissue. My last slide will be about the possible use. Actually, I would like to say thank you.
نشكر الدكتور هاني مرعي على التزامه بالوقت وعلى المحاضرة القيمة ونبدأ discussions في الزوم.
Thank you very much for the nice presentation. You spoke about if you destroy the cholinergic neuron, you make model by destroying the cholinergic neuron in animals, right? So what's the reason you use to confirm the cholinergic neuron already destroyed rather than the behavioral assessment? Like
as you know, we have assessed the generation of cholinergic neuron at the behavioral level, but now we studies are going to study the immunohistochemical differentiation of these cells. So this is the second part of our project, and also we will collect samples to test the gene expression profiling to see whether the cells are able to restore the cholinergic functions after the destruction and degeneration in ibotenic acid and xylitol.
And you already inject your neural stem cells in destroyed cholinergic neuron model, right? And you saw difference in the behavior of the nerve. But for like histochemical data or gene expression, not yet.
Okay.
Thank you.
تفضل.
Thank you, Dr. Hani. I wondered how you got the brain stem cell from your model. I think you sacrificed the animal. I'm looking for the future. How you expect you can collect the brain stem cell in the real life if you have a human application?
Actually, our neural stem cells have been collected during surgical brain operations in the Catholic University, Italy. But now we know through the directly programming protocol, we can easily transform somatic cell like a fibroblast using one transcription factor like SOX. So it's easy now to overcome the process of immune rejections. Exactly. No.
Thank you. Yeah.
What could be the difference between spinal cord stem cell and brain stem cell? Could it be related to the scaffolding behavior of spinal cord and stem cell, or what could be?
Actually, my expectation for this, why our cells have worked properly and the brain tissue in comparison to spinal cord, I think the niche itself, the combinations of neurotranscription morphogens and mitogens in the specific areas in the brain, the neurogenic areas in the brain are different from the spinal cord. So our cells, because they are isolated from the brain, they survive, grow, differentiate better in striatum and hippocampus, which are the same niche. Which route do you use to detect symptoms of spinal cord? So we have used a model spinal cord injury at the level of lumbar vertebrae 9 And this is not easy operation. We have made about 120 grafts. At the end of the experiment, I think we have only 20 grafts have survived this very difficult surgical operation.
The spinal cord is consists of gray matter and white matter. The white matter is formed of the tracts, mainly the permanent tract, descending from motor cortex to the motor neuron. When you cut the cord, the region immediately is cutting to the permanent tract, which is descending the action of the motor neuron in the area 4 and 6. So it is logic to inject cells to area 4 and area 6 better than the spinal cord itself.
Actually, we have no choice. We have made spinal cord injury. We have destroyed about 1/3 of the tract of the dorsal tract in the spinal cord, and you know how long this is a tract and the main lesion. So to restore this very long tract, it will be time. The time of our experiment was about 3 months, which I think is not enough to get the entire pictures and the entire regenerations of a very long nerve tract which are present in the spinal cord. And as I mentioned, there is a great difference between the compositions of the spinal cord tissue in comparison to the brain tissue. So, if we look for a better regeneration for the spinal cord injury, you have to isolate the neural stem cell from the spinal cord itself. And we know that the spinal cord contains some stem cells.
What I mean that we have the cell body in the area 4, the brain. Area 4 present in the brain, which contains the cell body. The axons present in the matter of the spinal cord. So, why not I inject the cells in the bacteria or in the area 4 and area 6 in the brain. So, it will generate the action as a prelude that tract descending through the spinal cord. And there, a clinical trial of human on autoimmune disease. They use red cells, clinical cells, and they inject it in the area 4 and area 6, and they got a good result.
Actually, this is a good idea. We didn't try it. You are talking about injections of neural stem cells in the brain itself and after regeneration, but as you know, this is a very long pathway, as it's not easy. And you know, after spinal cord injury, the spinal cord environment are very hostile for the growth of the cells. So, this is a very good idea if we inject the cells in the brain itself and we're looking for regeneration of the entire pathway. But this is theoretically possible. But practically, I think it's not an easy task. It's not an easy task because, as you know, the most difficult thing is the bad environment of the spinal cord following the injury. Very hostile astrogenesis occur, which prevents the growth and survival of most of neural stem cells.
You need regeneration, you need inflammation, and growth factor to get your regeneration.
As you know, our stem cells are genetically programmed to produce nerve growth factor, and we concentrating mainly on the trophic influence of nerve growth factor. I know that my cells maybe do not work, but I'm concentrating on the trophic influence of the genes that I have transfected into my cell line. And actually, previous study has disclaimed and reported success result and success story concerning the trophic influence of neural growth factor. Okay?
Thank you, Dr. Hani, for this lecture. I want to ask about what the expected risk factor in the future due to the different in genotyping between part of cells.
Our problem in the stem cells, whether it's neural or other type, is two problems. First, immunogenicity, second, tumorigenicity. And so, if we succeed to overcome these two problems, which is, of course, is not an easy task, so you have no problem with using or clinical application of stem cells. Up till now, the tumorigenic potential of stem cell, different types of stem cells are not overcome, so they are not safe. Most of the experiments are now at the preclinical level. Only few clinical trials are working now. For example, for the spinal cord injury, there are now, I think, one international company called Geo, something like that. They are working on the oligodendrocyte. They are injecting oligodendrocyte in a human spinal cord injury, and they are trying to assess the possible restoration of function following engraftment.
What about
the use of neural stem cells in diminishing the gene expression of abnormally phosphorylated tau protein and amyloid precursor protein, changing the structure of the protein from tertiary and going back to the amyloid precursor protein in normal amount in the transfected models of animal models of mouse in Alzheimer's disease.
You are reading my mind. Actually, I'm presenting a project now about the use of stem cells genetically engineered with the 3 genes which are responsible for dementia and Alzheimer's, which are amyloid beta genes and presenilin 1, presenilin 2. And actually, the evaluator of my proposal have an objection about this protocol. And they said if you succeed to renew the defective gene or the mutated gene, how about the old gene? They are still working in the background. So if you succeed to inject cells which are genetically engineered with wild-type genes, non-mutated gene, we still have the mutated gene working in the background. So I think we need to try this experiment. Actually, as I said, I'm working about this topic, and I hope I could get good results.
Already there is a gene is isolated from the neural stem cells, which is called seladine gene or selected in Alzheimer's disease, which is normal presence in the paraventricular area of the brain and showing gene expression of normal amount in age period before the age presentation of Alzheimer's disease, and then it is responsible for more deposition of amyloid precursor protein in normal amount that causes neurofibrillary tangents and forming the pathology of Alzheimer's disease. What about this gene?
Actually, this is not the only gene. You know, there are about more than 200 pathological mutations in the 3 main genes affecting the Alzheimer. And recently, there are more genes have discovered, about 6 or 7 genes. And as I said, the number of mutations about 200. So this is not only one gene. And as you know, that Alzheimer's disease is a very complex disease, and there is a massive destruction of cholinergic neurons, not only in the hippocampus, but in the different regions of the brain, and they include different areas of cerebral cortex, which control movement, control learning, which control most of the cognitive and the memory function. So it is not an easy task to search for just only a single mutation. Up till now, why Alzheimer's disease, what are the genetic basis? And I think the novel technologies such as next generation sequencing, we are working now on the next generation sequencing to try to find novel mutations in many numbers of genes. And this kind of study will require studies like genome-wide association studies and sequencing studies using next generation, which become very easy now and become cheap, and the time would certainly getting whole exome or whole genome analysis of individuals become affordable now.
Thank you.
Thank you. Now, the third one in this session, mesenchymal stem cells. Professor Elina Roms leads Institute of Stem Cells, United Kingdom, and welcome to you in our university and country.
Good morning. First of all, I would like to thank you for inviting me, particularly Professor Badawi. I'm very glad to be in Mansoura University and to be taught at your Regenerative conference. So my title of my talk will be about discovery of mesenchymal stem cell, the history and the current therapeutic use. Previous speakers very nicely described what the stem cell is. Just to mention again that defining feature of stem cell, its ability to self-renew and generate the identical cells. And the other feature, of course, is ability to generate the progenitor cells, which are less mature and able to form different lineages, as shown here for hematopoietic stem cell. So the history of mesenchymal stem cell begins with hematopoietic stem cell and the discovery of colony assays that allow people to enumerate regenerative cells. And this whole field opened up therapeutic applications in bone marrow transplantation in leukemias, and scientists were very interested about bone marrow microenvironment and resistance to chemotherapy. So now everybody recognizes that the father of mesenchymal stem cell is Alexander Friedenstein, and he worked in Russia in 1970s, also trying to see what cells are present in the marrow and can be transplantable. And he was the first one to find the second type of stem cell in the bone marrow, which were plastic adherent, well, in this case, adherent to glass. And they formed colonies of fibroblasts. What scientists in his lab did is they isolated individual colonies using cloning rings and transplanted them in diffusion chambers, which allowed migration of nutrients, but cells stayed inside the chambers, and implanted these chambers in nude mice. And several weeks afterwards, he was able to show that single-cell derived colonies of fibroblasts were able to form bone, cartilage, and fibrous tissues. Therefore, they fulfill the criteria for stem cells. The first scientist who recognized the Friedenstein work in the West was Arnold Kaplan, and he proposed this concept of mesenchymal stem cell, which was very similar to hematopoietic stem cell. At the top of the tree was a stem cell itself, and then he proposed the existence of progenitors of osteoblasts, chondrocytes, muscle cells, and many other various types of cells. And he called this process mesogenesis. Obviously, the fact that a single stem cell can generate all these lineages opened up great prospects for tissue regeneration. The next step in MSC history is this fundamental paper that was published in Science Journal in 1999. This was done by a company based in Baltimore called Osiris Therapeutics. And it's quite interesting, the name of the company, and I think it also relates to a myth of Osiris in Egyptian mythology, saying that you can regenerate the body of a person through a parts or stem cell. So these two scientists, they still work in the field, and they published this famous paper on mesenchymal stem cell characteristics. They will show that these cells are clonogenic, able to form colonies, and they developed in vitro assays for multiple potentiality, which is much easier than in vivo assays. This is assay for adipogenesis, osteogenesis, and this is assay for chondrogenesis, in which cells are grown into little balls, which forces them to form cartilage tissue. There are also a number of markers, CD markers was proposed that are specific for cultured mesenchymal stem cells. Straight after this publication, we can see clinical trials emerging with mesenchymal stem cells. It's this one, the first one, which was performed in Italy and Russia. 3 patients were used with very large segmental bone defects. And in this study, mesenchymal stem cells were expanded from the bone marrow and then seeded on scaffolds that were the size of the bone defect and fixed. And the first results were published in 2001, showed very good results, and 7 years outcome results were also excellent. This shows the ability of MSCs to form bone. These are other indications in which bone marrow MSCs can be used. For bone fracture, we use it in leads, also in dentistry, spinal fusion, and also building bone after rejection of tumor. The clinical trials now take place for osteogenesis imperfecta, also osteonecrosis of the femoral head, non-union fracture. There's many clinical trials now happening with bone marrow mesenchymal stem cells. The other very important study I would to mention to you is a study by the Swedish group, Katarina Laplan. They were the first one to show the mesenchymal stem cell, in addition to their ability to differentiate, they also have very strong immunoregulatory function. They are able to dampen immune responses. At the time, the mechanism wasn't known, but as a result of this very interesting function, there's now many very successful clinical trials in which MSCs are used to inhibit immune responses. And particularly successful, there's a trial for graft versus host disease, but also for diseases like Crohn's disease. These are just the current trials for graft versus host disease. As you see, some of them conducted by Osiris Therapeutics, but also by other centers in the world, and are very successful. This is just a diagram showing that Osiris Therapeutics are still very active, but they are mostly interested now, not in bone and cartilage repair, but in graft versus host disease repair, Crohn's disease, and also repairing heart attacks and cardiac tissue repair. I'm not an expert in cardiac repair because I work in the rheumatology field, but I just want to mention one recently published trial which used autologous bone marrow-derived mesenchymal stem cells, and they were delivered by injection. It's a phase 2 study, and they show very satisfactory safety profile. They also show improvement and structural and functional measures. At the moment in the cardiac field, it's not quite clear whether the mechanism of action is simply differentiation to muscle cell or some other factors are involved, such as immunoregulatory activity, and also the ability to support resident cardiac stem cell is also important. This, as I mentioned, MSC is not simply differentiate, they also produce very many useful cytokines and growth factors that support resident stem cells and vasculature. Some of them I'd like to mention is stromal cell-derived factor 1, which is the chemokine that promotes migration of resident stem cells to the repair site. These two are immunoregulatory cytokines, and of course, VEGF is the factor that is important for bringing vasculature to the repair site and therefore promoting rapid regeneration. This is this is the last year snapshot of clinical trials that currently take place using mesenchymal stem cells. And you can see the majority are still dedicated to repairing skeletal tissues, bone, muscle, and cartilage. But heart diseases picking up the pace, also the digestive diseases, nervous system diseases, as previous speaker mentioned before. In some cases, mesenchymal stem cells also used for spinal cord injuries and multiple sclerosis, the cancers, graft versus host disease, and also metabolic diseases like diabetes and connective tissue and skin diseases, including lupus. So you can see that the range of diseases that now being tried to treat with mesenchymal stem cells is very broad. And this is just the overview of clinical trial as per this month. And you can see that quite a lot of trials happening in the US and in Europe, but interestingly, many trials happening in China, Far East, and the Middle East as well. So it would be very nice to establish collaborations and potential trials with your university. Just briefly, what do people do when they use mesenchymal stem cells? Normally people extract mesenchymal stem cells from the bone marrow by aspiration. They process the sample, they isolate cells, put them in a flask to derive adherent fraction, then expand cells using trypsinization, and they get billions of cells. And then they centrifuge cells and use as therapy, either inject into bone fracture or seed on the scaffold And this is one of the paper in which MSCs were found in synovial lining tissue. And the cells were found particularly good in repairing cartilage. The other interesting tissue, which is very popular these days, is adipose tissue. Again, in 2001, paper by Zook found MSCs in adipose tissue, and they showed that the cells were osteogenic, chondrogenic, all the rest of it, but the frequency of cells were much higher than it was in bone marrow. And this again opens up avenues for therapy, and in many cases, MSCs now are obtained from lipoaspirates. This is a very rich source of MSCs. There's a company in America, Cytori, who make these products of MSCs based on lipoaspirates. And of course, advantage is that there's no shortage in donors, and obviously the cells are no problem to obtain. Another interesting source which became very popular is umbilical cord. Again, it's a waste product, and MSCs were found in umbilical cord in 2003. Frequency is very high, cells are very proliferative. Again, many companies in the world, including this Korean company now making products based on umbilical cord MSCs, and particularly used for neuronal tissue regeneration. We worked in with these cells in terms of bone repair, and we directly compared them with bone marrow MSCs using these assays for regenerative bone. As you can see in our hands, we couldn't find umbilical cord MSCs as good as bone marrow in forming bone. So for our interest, umbilical cord cells may not be that potent, but they may be good for neuronal regeneration. We also first to find MSCs in synovial fluid, not in tissue, but in fluid, and we found them in patients with different types of arthritis. We compared them with bone marrow MSCs, and we found that actually synovial fluid MSCs are much more proliferative, and they're also more chondrogenic. If you compare bone marrow MSCs forming cartilage on scaffold with synovial fluid MSCs are better. And also found MSCs in periosteal tissue, and showed that periosteal MSCs are better in forming bone than synovial cells. So what I want you to take out of this presentation is, when you're thinking about using MSC as therapy, you need to think about three different approaches. First of all, you need to decide which source of MSCs you would like to use for particular disease. Because MSCs, although they termed MSCs, they are slightly different in different tissues, and this depends on what they naturally do in this tissue. So once you've decided on what sort of source of MSCs you're planning to use, the next thing to think is when to harvest. And I'll just briefly explain it on the next slide. And the final question to decide is how long you're going to grow your cells. So when to harvest? We did some work on synovial MSCs, and it directly relates to the level of inflammation. So obviously, if you want to treat patients with rheumatoid arthritis, it's not a good time. The first thing you need to do is reduce inflammation and then harvest MSCs, but not the other way around. Only briefly about cultured MSCs. In most clinical trials, cultured MSCs are used. As I say, many flasks cells are cultured through. But it is now well known that the longer you culture MSCs, they lose their proliferative potential and also differentiation potential. You can see here the aging culture, they lose their telomeres, and they also acquire markers of senescence and potentially also genetic abnormalities. So in practice, all manufacturers of MSCs trying to use MSC as a very low passage. Of course, you can get many cells at late passage, but cells are not as good, they're not as proliferative, and maybe potentially even tumorigenic. Just a very brief couple of slides. As we now know, MSC is present in many tissues, but where are they actually located? People still don't know what sort of can actually be described as MSC, what sort of markers it has. So the most current concept of MSC topography is that of a pericyte, because in most tissues we have vascular system, and vessels are supported by these cells called pericytes. And currently people believe that these are the pericytes that support blood vessels, but it can also be organized and activated to become an MSC. However, we don't believe that pericyte concept explains all the MSCs, because MSCs can also be found in fluids where there is no blood vessels. For example, in synovial fluid and umbilical cord blood. So we think maybe in these cases MSCs are shed from the blood vessels onto the fluids, but they still exist in the fluids and are very important. And this brings us to understanding that there can be some other ways of using MSCs without expansion at all, by understanding what MSCs actually do in fluids. And this is the one work that I did with University of Columbia in New York, in which they tried to repair cartilage using MSCs, but not actually without doing any expansion at all. So what they did is they created a defect in the rabbit joint, and then they put the scaffold that was soaked with chemoattractive growth factors. And after some time, they actually found that the scaffolds were colonized by cells and created excellent repair. So their concept is actually, rather than expanding MSCs and injecting them back, trying to use the potential of native cells, so it is believed that this repair is due to MSCs present in synovial fluid. So the scaffold is able to attract MSCs, and the other growth factor in the scaffold was able to produce their differentiation. So it's just a different way of thinking about using MSCs in clinic, not simply by expanding them, but also by attracting them and forcing them to differentiate the desired lineage. So at the end of my talk, I'd just like to conclude, and I'd like to take some messages back home. First of all, MSCs were discovered in the bone marrow, but they found subsequently in almost every single adult or even perinatal tissue. They have multi-lineage differentiation potential, but they also have multiple trophic effects, including very strong immunoregulatory capacity. Based on these functions, they've been used in many diseases, including spinal cord and other neurological diseases. And best source of MSCs for individual disease should be found. And how MSCs are cultivated for how long is also important to predict what therapeutic outcome they will bring. And in the end, I'd just like to thank you, my lab. We're based in Leeds. This is our funding body. We're very lucky to be funded by European Community, also several industrial partners. And on the left are my collaborators from Aberdeen, from Columbia University, Sheffield University. These are the people who made the scaffolds. And Frederick von Schell, also my colleague in Leeds. Thank you very much for your patience.
Thanks so much to Professor Zelina Jones, and now the discussion on floor. Anyone?
Thank you, Dr. Zelina. Being an expert in the mesenchymal stem cell, do you believe in your heart that the bone marrow mesenchymal stem cell differentiate totally and with total function to different tissue, or it just paves the land for the organ stem cell to be activated at the very level?
If I understand your question correctly, in the bone marrow, about bone marrow MSCs, yes? And bone marrow MSCs are tripotential at least, and they are good at forming bone, but also they have immunoregulatory capacity that is probably related to their strong function in bone marrow supporting hematopoiesis, because hematopoietic and mesenchymal stem cells exist together. So, if you to use mesenchymal stem cells from the bone marrow, they will be good for bone regeneration as well as for immunoregulatory capacity. Is it the right answer?
Yeah, I'm extrapolating the data from repair of the heart, for example, by having a function. We know that now that mesenchymal stem cells doesn't change into cardiac muscle cells, but they are just making a good environment for the cardiac stem cell to repair.
It's absolutely true, yeah. The first trials were initiated based on the idea that MSCs can differentiate into myogenic cells, which they do. But the problem is they don't exist very long in the heart, they disappear. So based on this, people decided, worked out the mechanism of action is more likely to be supporting of resident cardiac stem cells, supporting vasculature, also promoting cell survival, reducing apoptosis. So there's many functions which people don't understand at the moment. But as soon as they understand what MSCs do in the heart, they will probably just use the cocktails of growth factors instead of MSCs themselves. But it's not known at the moment because it's a multiple functions.
My question is not so far from the same question. The mesenchymal stem cells for cardiac repair, you mentioned that something. As I know that mesenchymal stem cells, many people recently, we've read about that mesenchymal stem cells, okay, can differentiate to cardiomyocytes, and when they transplant, it works, but not fully functional. For example, they found that coenzyme 43 unit secretion is active in case of cardiomyocytes come from mesenchymal cells. And this causes arrhythmia after treatment. So, and they, as I read, they said that it's not preferable as a source for cardiomyocytes.
Yeah. Yes.
I'm not sure.
Is this about human trials?
Human trials, yeah. Many trials on cardiac repair.
And is that comparable to the CD34 or CD133?
I think most people use 133 purified bone marrow hematopoietic stem cells.
Was it Clinimax?
Yeah. As far as I know.
This one is entirely using Clinimax?
No, these are fully cultured expanded plastic adherent cells. Okay?
Cultured cells?
Yes, yeah. As far as I know, there hasn't been a clinical trial which directly compared CD133 and MSCs. That hasn't been done. So it would be very interesting to see if cultured mesenchymal stem cells comparable to CD133 cells. Would be very interesting to see.
Is this part of the study?
Yeah. Or some clinical trials, phase 2 trials are completed now. As I showed, the results are published. So definitely MSCs are safe. That's phase 1 trials. And phase 2 trials show some improvement in cardiac function. I don't work in cardiac field. So, I only know what I read. And people now in cardiac field are very interested in using mesenchymal stem cells, whether they would be from the bone marrow or from the other tissue like umbilical cord or adipose tissue. It would be very interesting to compare. But because it's such a big market, then these trials are taking place.
What about the dose?
The dose of MSCs, I think it's 10 to the 8 per kilogram of body weight. 100 million cells per kilogram. It's a huge dose. Some of them dose escalation studies, they're using less dose, but it's a huge dose. It means that cells need to be expanded controlled. But some trials show that allogeneic cells are effective. So if people can use allogeneic cells, this is no problem.
What's the route of injection?
Various. Some use systemic injections, some use intracardiac injections, and the different parts of the heart, but I don't quite remember.
Thank you, Dr. Ray.
Thank you. Is there a difference between the kind of stem cell isolated from the core tissue itself and portable?
Okay, this is a very interesting question because first studies found MSCs in umbilical cord blood, like hematopoietic stem cells, blood. MSCs were found in blood. They were very, very rare, but similar in growth to bone marrow MSCs. But they were very rare. Sometimes, maybe only 20% of donors' blood could grow MSCs. And we tried as well, and it's unpredictable. But later on, people found that actually they can grow MSCs from Wharton jelly, but either by simply squeezing cords and just squeezing them and pulling blood. By doing this, you get more MSCs. So people understood that MSCs might be present inside the cord, and they digest the cord tissue with enzymes like collagenase, and they found that inside the cord itself, there's 1,000-fold more MSCs than an umbilical cord blood. So actually, they're present inside Wharton jelly. And the only reason they're present in cord blood is because of squeezing blood vessels and physically pushing cells into the blood. So if you're thinking about using umbilical cord tissue, I would recommend using umbilical cord rather than umbilical cord blood because it's much more cells in the cord itself.
As general talk, what rules the differentiation of mesenchymal stem cells into different cell lineages?
I can't see asking the question.
What rules the differentiation of mesenchymal stem cells into different cell lineages? And what guard against tumor genesis in this process?
I understand the second part about tumor genesis. I don't understand the first part.
What sort of lineage you can get from mesenchymal stem cells?
What rules? Are there certain growth factors that rules the differentiation of the mesenchymal stem cells into different cell types? Neural stem cells, chondrogenic stem cells, and so on.
Well, yeah, of course, there are different chemical agents and growth factors that people can use to induce differentiation. For example, for cartilage, people use TGF-beta. For adipogenic, people use things like insulin. So there's a various media formulations to drive MSCs to different lineages. And lineages are fairly stable. In terms of tumorigenicity, yes, it's a very big problem or concern because in mouse experiments, scientists found quite a lot of chromosomal abnormalities when they grow mouse MSCs, and in some experiments, tumor formation. However, this never happened with human MSCs. Not a single clinical trial reported safety concerns, even though some chromosomal abnormalities sometimes can happen, but never tumor formation ever been recorded. So, so far, MSCs are proven to be safe. But as I mentioned, I would not recommend growing MSCs for very long, because the longer cells are grown, the higher potential to form, to create any abnormalities.
Thanks, Madam. I would like to ask, as you know, spinal stem cells can differentiate to many types of cells like muscles and fibrocytes. In certain cases like cardiomyopathy also applied for a muscle, differentiation of heart and brain. How can I expect that stem cells will all differentiate to fibrocytes? And this is what I don't like talking. So,
correctly what you're saying, if we inject undifferentiated MSCs, how can we make sure they go along the desired lineages rather than all the rest of it? Yeah? This is a very good question. That's why I'm a bit concerned about systemic injection of MSCs. I would rather, in our group, we prefer to seed cells in scaffolds or inject them locally where the defect is. And the defect environment itself creates the cytokine mutually to induce the right way of differentiation. So I am concerned about systemic delivery as you are, because it could be some adverse events. But if you inject cells locally, and particularly with the cocktails of growth factors, like in Colombia's study, that specifically drive cells to differentiate, this is a much better approach.
Okay, I'm just asking about your own work on osteoarthritis. It would be a role model for regenerative medicine where it is actually degenerative. You can take cells autologous from bone marrow. You can inject it locally through arthroscope. So ideally, this would be the optimum or the best example of regenerative medicine. And again, in aging, a very common disease. Yet, there is no breakthrough except the one clinical trial that you referred to. From your point of view, as an expert in the field, how long is it going to take that ideal model of regenerative medicine to work based on the current status today? Thank you.
This is again a very, very good question, and I take your point about there's no major breakthrough in terms of cartilage regeneration. That's what you mean. I think cartilage regeneration, I mean, there's a big success in bone regeneration, definitely. You know, even in osteogenesis imperfecta, osteocrostohumeral head, but cartilage is much harder to repair because it's avascular tissue, and also it's bathed in synovial fluid, which is anti-adhesive. So the biggest challenge in cartilage repair is to make sure the cells are present in defect, they stop there, and they differentiate. So the way I'm thinking about it, cartilage repair, is using scaffolds, using some matrices to make sure the defect is completely separated from the fluid and the cells are retained there and forced to differentiate. So in terms of cartilage repair, I think the way forward is scaffolds that place the growth factors and also with MSCs, whether they are culture expanded or freshly isolated and purified. Future will show which one is the best way, but scaffold is most important in cartilage repair. Thank you very much for your nice questions.
Thank you for Professor Edina, and now the last speakers, Professors Professors Assam Abdel Alim, Assistant Professor Cytology, Suez Canal University, embryonic stem cells.
Thank you very much for inviting me today to be here in Mansoura University. Today I will speak about some basics of pluripotent stem cells, particularly embryonic stem cells and induced pluripotent stem cells. First, what stem cells? Stem cells is a cell that has the incredible ability to replicate or self-renewal and differentiate into different cell types. General characteristic of stem cells: it's undifferentiated cell and specialized capable of dividing or self-renewal and able to differentiate into several cell types. Stem cell potency. In the beginning, I would like to speak about the different potency of different types of stem cells, because not all stem cells have the same potency. So a multipotent stem cell, the cells that can give rise to several cell types, but not all cell types. And the example of this cell is adult stem cells, and pluripotent stem cells, the cells which can give all types of cells of the body, and also extra embryonic tissue. And the good example for this is embryonic stem cells and induced pluripotent stem cell. And totipotent cells, these cells that can give complete individual when implanted into uterus, like zygote, and give complete individual. Different type of stem cells. The adult stem cells, which we said is they are multipotent, and embryonic germ cells. Sure, adult stem cells, including several types like hematopoietic stem cells, mesenchymal stem cells, neural stem cells, umbilical cord stem cells. That means tissue-specific cells or somatic cells. Embryonic stem cells, embryonic germ cells, the cells which derived from the primordial germ cells during late embryo stage, and it's considered pluripotent. And embryonic stem cells, we will speak today about the embryonic stem cells and diffuse pluripotent stem cells, which originate from the first week after fertilization. And induced pluripotent stem cells, which come from somatic cells. Embryonic stem cells. What is the source of embryonic stem cells? In human, we can obtain the embryonic stem cells from two sources. Eggs, fertilized eggs from IVF clinics, embedded fertilization clinics, on therapeutic cloning, somatic cell transfer. Sure, this in Egypt is impossible. But eggs, fertilized eggs, it's possible, I think. To know that where embryonic stem cells come from, let's speak generally about embryogenesis. After fertilization at day 1, the zygote is formed, then division happens at 2-day cell stage, and then 4th cell stage, then 8th cell stage, then normal stage, or multi-cell embryo. After that, blastocyst formed at day 5 to 7 days. All this stage, before this stage called pre-implantation embryo, and then the embryo implanted in uterus, and the tissue different tissue start to be formed. Where embryonic stem cells come from? Embryonic stem cells come from blastocyst. A blastocyst consists of two parts, outer part, which true them, which will form the placenta, and the inner part, which called the inner cell mass, and this will give fetus in the future after implantation. So we take this part, we isolate the inner cell mass, and that means we destroy the embryo, and then we culture this inner cell mass in tissue culture plates. Before we culture these cells, we prepare plates with certain types of cells, and we'll speak later, or we prepare specific type of medium, which is very important to be specific for the type, depending on the animal type. If we think about human or mouse or monkey or whatever. After that, when we culture these cells, we propagate in vitro, because these cells have incredible ability to proliferate. So we make like millions of cells. Then after that, if we decide to go to certain lineage like liver or neurons or muscles, we apply different protocol according to our target. Sure, our last target is cell therapy. We give special properties for embryonic stem cells. What's embryonic stem cell pluripotent, as we said, able to form any cell type of the body. So it's the most powerful stem cell. Second, they have incredible ability for self-renewal and express the enzyme telomerase. That means the cells cannot be aged. And also express very specific markers like OCT4 and other markers, which gives the strength for this pluripotency. So OCT4, if the cells maintain certain level of OCT4, cannot be differentiated. But if the OCT4 down-regulated, these cells switch to differentiation. And also, these cells have the ability to maintain normal chromosome structure, even after very long time of culture. And this characteristic is different from several types of cell lines, other cell lines. And embryonic stem cells have the ability to form teratoma. When we inject embryonic stem cells in the nude mouse, immune deficient mouse, we expect to have teratoma. When we make histological section, we should have all lineage like mesoderm, endoderm, ectoderm. this kind of marker for pluripotency. So embryonic stem cells have the incredible ability for self-renewal, and to keep in culture, these cells must be maintained pluripotent. How to maintain these cells pluripotent? We need to suppress differentiation of the cells and to enhance the proliferation. To be maintained pluripotent, because if the cells are not maintained in pluripotent state, we cannot differentiate to appropriate cell type. For this, the self-renewal and the pluripotency regulator are responsible for both stimulation of proliferation and inhibition of differentiation. There is two types of signaling mechanism. Exogenous signaling mechanism, endogenous signaling mechanism, that means the embryonic stem cells themselves, they express several genes and protein which maintain these cells pluripotent. Most important of these genes, there are several genes, like OCT4, NANOX, SOX2, CAL4. This is very essential to maintain the cells pluripotent. If any gene from this genes down-regulated, the cell automatically differentiate, and we cannot maintain culture. And also, there is exogenous signaling. Exogenous signal, we have to add this signal in culture, during culturing of the cells. In vitro, for example, like leukemia inhibitory factor, in mouse embryonic stem cells, if you not add leaf or leukemia inhibitory factor, you cannot maintain the cells pluripotent. And also, like basic FGF in human A cells, it's essential to be added in culture. So this factor, we supplement this factors in the culture media. This example for human A cells, as we know that human A cells first discovered 1998 by Thomson. And this is the IEI colony grow on feeder layer. This feeder layer originated from mouse. It's not from human. Monkey embryonic stem cells and the human embryonic stem cells have similar characteristics for gene expression, and even for maintaining the cells in culture, they require the same requirement. For example, to be maintained pluripotent, we need fibroblasts. This fibroblasts come from embryonic mouse fibroblast. We kill the mouse and we obtain that at 12 to 14 days, and then we remove the head and the neurons and blood, and then we make primary culture, then blade this fibroblast and inactivate by some anti-cancer drugs like mitomycin C, and then we blade the colonies. And now, because this is very important to exclude the feeder, recently we can culture human and monkey embryonic stem cells without feeder layer, and this is very important. Mouse embryonic stem cells is more strong because discovered 1981, and the research in mouse embryonic stem cells for a longer time, and now we can culture very easily without any feeder layer, and sure it's more strong. This example, how or protein expression of some markers in undifferentiated embryonic stem cells. This from our work published this year. The first part, this embryonic undifferentiated colony from murine embryonic stem cells express OCT4 markers, which is present in nucleus, and we discovered the new markers or new factors specifically in undifferentiated stem cells, the NBRC. So to make sure this is really expressed in undifferentiated stem cells, we use OCT4 as a marker. And we make colocalization by confocal microscopy, and we find the same cell which express OCT4 express NBRC. To make sure this is true, we turn back to where embryonic stem cells come from. Embryonic stem cells come from blastocysts. So we turn back to in vivo, and we obtain blastocysts. In case of mouse, the blastocysts at day 3.5. So we obtain the blastocysts and we stain the immunocytochemically, and we find same that NBRC expressed in OCT4 positive cells. That means inner cell mass, the source for stem cells. And also we make several examinations like gene expression and protein expression using Western blotting, and we find it is the same result. So that means that this new factors is really expressed in undifferentiated condition. We'll leave embryonic stem cells and we'll return it back, and we'll speak about induced pluripotent stem cells because both are very close. Because without embryonic stem cells, there is no induced pluripotent stem cells. Induced pluripotent stem cells, it's a combination, and this is discovered 2006 and 2007 by Professor Shinya Manaka, Japanese professor. And he depended on the fact that embryonic stem cells can express cell renewal or pluripotency regulators like OCT4, NIC, and several factors. He chose 4 factors, the most powerful. He thought this is the most powerful factors or the transcription factor, and he said how we can put this factor inside somatic cells that already differentiated, like skin cells or fibroblasts, whatever, and see what happens. So he made a combination of several genes, and he finds that they can reprogram somatic cells or already differentiated cells into pluripotent cells. That means these somatic cells that already differentiated return back to embryonic life, embryonic state. And this process called the reprogramming process. And he used 4 factors, and he transcribed this factor using a viral vector. So, and this pluripotent cells or induced pluripotent stem cells they become like embryonic stem cells. Same characteristics, same gene expression, same morphology, same epigenetic status, same in vitro differentiation system. This Professor Shinya Manaka and his co-worker Takahashi, Dr. Takahashi, they published in Cell 2006. This is a big paper, and he got Nobel Prize for this paper this year. He made induction of pluripotent stem cells from mouse, embryonic, and adult fibroblast culture by different factors. He made primary culture from fibroblast. Then he, by viral vector, he transfects 4 genes, and he found after that he found that this cells fibroblast converted to pluripotent cells and have stem cell characteristics. And in 2007 he made same for human, and also published in Cell induction of pluripotent stem cell from adult and human fibroblast by defining factor. And this is the morphology of human IBS cells. Why did he say to make IBS cells? Because he to resolve the problems that come from embryonic stem cells. Embryonic stem cells.
Please, the time 2 minutes and 5.
Okay, let's speak about advantage and disadvantage of induced pluripotent stem cells. Induced pluripotent stem cells come from patient and return back to the patient, so genetically identical to patient. We no need to destroy embryo and pluripotent. Disadvantage of our problem still we have with induced pluripotent stem cells that still have genetic effect and use of viral vector, but now already they can induce pluripotent stem cells without viral vector. This year, Nobel Prize for Physiology or Medicine 2012 was given to Professor Shinya Manaka and Professor Sir John Jordan for the discovery that mature cells can be reprogrammed to become pluripotent. Let's speak about the Nobel Prize winner. So Professor Sir John Jordan, he got Nobel Prize this year because he could clone in 1962. Jordan successfully cloned a frog using nuclei from somatic cells. 1962, and then Shinya Manaka came in 2006, and he successfully produced embryonic induced pluripotent stem cells that may convert somatic cells into induced pluripotent stem cells, depending on the idea of John Jordan. So I will not speak too much about this slide. So just I will show some differentiation from our work. For example, we can differentiate into heart cells. Sorry, the video is not working. I don't know why. Anyway, we could obtain beating cardomyocytes in the culture. So these two videos are beating cardomyocytes in culture, in vitro. And after that, we could purify this culture from cardomyocytes and propagate, depending on the fact that in prenatal life, the cardomyocyte can make hyperplasia, but after birth, there is no hyperplasia, only hypertrophy. So depending on this fact, we could purify and propagate the cardomyocytes into several colonies beating in the culture. This phenomenon is very important when we want to make transfer, because we need large number for cardomyocytes for transplantation. Second, the risk of tumor formation or teratoma, because any contamination with differentiated cells with undifferentiated cells will lead to the teratoma. Also, we show here that we could obtain functional cardomyocytes in vitro. Like, for example, we know that histologically sarcomeric organization is very kind of functional structural unit of cardomyocytes. So we could obtain cells which already have sarcomeric organization in vitro. We show here that we can differentiate embryonic stem cells into neuroglia and the glia cells like neurons, the general neurons, or astrocytes or oligodendrocytes. Peripheral stem cell therapy, not embryonic but generally, only disorder of blood, disorder of immune system, loss of bone marrow function, skin reaction. For stem cells, embryonic stem cells, only successful treatment for animal models. Severe immune deficiency, diabetes, Parkinson's, spinal injury, demyelination, myocardial infarction. Only recently one case from human embryonic stem cells were treated and were published in Lancet 2012, and the two patients with macular degeneration, and they produce retinal pigment epithelial cells, and they could transplant this retinal pigment epithelial cells into the patient, two patients, 55 years old and 75 years old, until publishing this paper, it was 7 months after transplantation, and they started to see again. A challenge for stem cells or embryonic stem cells especially, need to be differentiated to appropriate cell type because they can't be used clinically. Stem cell proliferation must be controlled once placed into the patient. Possibility of rejection of embryonic stem cells is very high, and the contamination also possible. The use of feeder to grow cells could result in problem. Thank you very much. I'm sorry for this.
Thank you, Dr. Tom. And now only one or two questions in this part. One or two questions. Any questions? Okay.
May I ask you what is the future of work on embryonic stem cell in the era of the future? Do you expect that to be proceed?
As you see here, we already have clinical trial, one clinical trial in human, and published in Lancet that they already treat macular degeneration. And also, you know, the history of embryonic stem cells is very short. Embryonic stem cells, the human embryonic stem cells discovered 1998. So clinical trial of embryonic stem cells just started. And also, without embryonic stem cells, there is no IBS. That means, without research in embryonic stem cells, we cannot get IBS. So I think both will go.
Okay.
Embryonic stem cells, there is no debate about that the embryonic stem cells are the most powerful stem cells because it is a fetus. It is the fetus, so that theoretically must give everything or should give all cell types. So no problem. But the problem for embryonic stem cells is immune rejection. That you cannot transplant embryonic stem cells as undifferentiated, because if you transplant as undifferentiated, we cause teratoma or cancer. But you have to differentiate to appropriate cell type, fully differentiated without contamination with other type. And when you transplant, immune rejection happens. So that's why they, IBS treat this problem or just patient specific. They'll take somatic cell from patient and return it to the same patient. But this cycle takes years. It's not just very simple. Take like 1.5 years. For example, all blood diseases already proven to be treated by adult stem cells. So in this case, I think no need to think too much about all blood diseases and leukemia already proven. But we all scientists now they try to treat other untreated disease. So all different types they try different types. But who would succeed? We don't know.
Finally, thank you very much, and I would like to thank the chairperson, Dr. Mohammed Youssef, the Dean of the Faculty of Veterinary Medicine, the Drs. Azza Ismail, Professor Dr. Azza Ismail, Vice Dean for Research and Graduate Study, Faculty of Science. Also, I would like to thank all the speakers and attendants and the chairman of this conference. And I hope that the sessions will be successful and will open in the future several fields and areas in the research, in advanced research concerned to these sessions. Thank you very much for all. The next session will start at about after 5 minutes. The opening ceremony to this conference.
بسم الله الرحمن الرحيم. وقل اعملوا فسيرى الله عملكم ورسوله والمؤمنون. تتشرف كلية طب المنصورة بإقامة المؤتمر العالمي لأطباء التجددي تحت إشراف الأستاذ الفاضل الأستاذ الدكتور محمد صبحي ومشاركة جميع أعضاء تأسيس الكلية والسادة الزملاء والعمداء من كليات الطب الأخرى وكلية الطب البيطري وكلية الصيدلة. وسيقوم الأستاذ الدكتور محمد صبحي بإلقاء كلمة الافتتاح. ونرحب بصدركم جميعا بإلقاء الدكتور محمد صبحي.
طبعا في البدايه واشكر سياده الدكتور سيد عبد الطالب رئيس الجامعه والاستاذ الدكتور عماد نصر والاستاذ الدكتور جهاد سعد لتشريفهم ودعمنا المستمر سواء في مركز التدريبات التجريبيه او في المستقبل القادم فيما يخص مركز الطب التجريبي لجامعه المنصوره. في البدايه أحب to start with, I have to present this, that the laboratory dealing with human physiology is the most complicated laboratory, and so it needs the most and the greatest attention and care. And I will talk a few words about the objective of our center. We look for development of experimental and scientific research in Faculty of Medicine, Mansoura University. We look for raising awareness of the importance of empirical research among under and postgraduate students, faculty members, and assistants. We are looking for opening up the world of scientific research, cooperation between University of Mansoura and other universities and scientific research centers both in Egypt and outside Egypt. Also, we are looking for scientific cooperation with international scientific centers. The areas of integration and compatibility between the basic research disciplines and the interdisciplinary clinical faculty of medicine in Mansoura University, one of our major objectives. Also, the implementation of workshops for different departments, and the implementation of local training courses and international empirical research for young researchers. So our mission, MERC mission, is to foster research in the field of biomedicine inside Mansoura University, and our vision is to be ranked as the first center for biomedical research in Egypt and Arab world. Now, a look for the research being done now in our center. MERC supported biomedical researches with a special emphasis on the stem cell technology, which is the research plan of Mansoura University. Different types of researches are being conducted in our center. Now we have PhD theses, we have grants, national and international. Also, we as MERC supply or made it possible grants for medical students, undergraduate medical students. Also, we support free research for the staff members. Now we come to the PhD thesis. Over 20 PhD theses are being conducted now in MERC. Candidates from different faculties came to MERC seeking for support for their thesis, from Faculty of Medicine, Dentistry, Science, College of Science, and Pharmaceuticals. Also, in our center, we have grants, national grants. We have two Mansoura University competitive grants, research grants, and we have 7 STDF grants. Also, we have one international grant between Egypt and Germany on Parkinsonism. Regarding one of our very important, we think one of our successes is the MERC student summer grant. Since in 2010, MERC began an ambitious plan to integrate medical students, and medical students, I mean undergrad, into research, and this was the first cycle. In this first cycle, 7 accepted researchers in international conferences. From our student team, we have 7 research projects. All these research projects have been accepted for presentation in international meetings, and 2 of them were awarded the best research, one in neurology and the other in nephrology. This was in a conference in Holland. And also, in the first conference by the Minister of Higher Education, student research, 4 out of the first 5 places were awarded to student research from MERC. Out of these student research protocols, one has been accepted in an international journal. This is the CNS and neurological disorder. This is a journal with an impact factor of 3.8. Now, in this year, in the second cycle, we expanded the scope of our project to involve all biomedical students from Mansoura University. We included medicine, beside pharmacy, beside dentistry, and College of Veterinary Medicine to stimulate the teamwork in research. And now we have nearly 16 research projects, and students are very enthusiastic in doing this. After the enthusiasm for application to the second cycle this year, we believe that this program research reached its goal. Now we have students competing for making research, which is very important for our community. Regarding free research, MERC is also devoted to support Mansoura University staff need to perform their research projects helped by our equipment, staff members, and expertise. About sections in our institution, we have an open animal facility. MERC contains a big facility for experimental animals, including places for mice, rats, rabbits, dogs, and with different strains. This place is being directed by a group of experienced veterinarians. Also, recently, we are supported by our team, and now we have a closed animal facility specified for care of special strains of animals, transgenic and immunocompromised mice. Regarding also, we have 2 surgical rooms, well equipped for animal operation. And in this animal theater, we did coronary bypass and also liver transplant and experimentation. We have tissue lab, tissue culture lab, and this is one of the strengths of MERC. In this unit, with a special experience in different types of stem cell and their application. Also, we have a molecular biology lab for different genes analysis and characterization. We have pathology lab where all equipment necessary for tissue processing, cutting, staining, and imaging analysis are available. Also, and we are proud by this, as a result of collaboration with our center and the German center, we have now an experimental neurology unit, which is fully equipped with video tracking system necessary for behavioral assessment of animal model. Moreover, a complete stereotaxis system is available for intracranial injection. Now, regarding international collaboration, one of our strategic goals is to build a powerful collaboration with international centers of excellence all over the world. Example of such center is one center in Germany and other center in the United States. Not only research in our center, we do other things. We do training, we do teaching for students, and we do communications, and also we have our dream, which is now a real, which is the about the real. This is the Regenerative Medicine Master Degree, and also Regenerative Mansoura University Regenerative Medicine Center. Regarding training courses for staff members and students are carried out, we have workshop stem cell physics. We have also workshop for microsurgery, vascular surgery, and experimental animal training workshop. Teaching, we are proud that medical students are receiving a course in research methodology in MERC to teach them principles of biomedical research. Communication, MERC hold an annual international conference to help communication with different international research schools. Also, the main research aim, which is stem cell, the study we established in our center, stem cell application. We have PhD theses and grants for using stem in different organs, brain, heart, kidney, and liver. And this is one of our international publication, and we have many like this. Regarding the regenerative medicine master degree, the regenerative medicine master degree is a degree with a credit hour system aimed at supplying candidate with the needed skill for conducting research in the field of stem cell, and this degree we are doing our best to be real, and we are about to achieve this. Also, we are about to achieve our future goal for Mansoura University, which is the Mansoura University Regenerative Medicine Center. And this center, comprised of the core present in Mansoura Faculty of Medicine, and our dean supplied us with a place for this core, in which medicine, staff members in Faculty of Medicine, meeting with the scientists from College of Science, Pharmacy, Dentists, and Veterinary. They meet here to do the basic research related to stem cell and related nanotechnology practice. And attached to this, a clinical working group in different departments in Mansoura University Hospital and medical centers for actual application on human beings. On celebrating the launch of our program, MERC organized the annual conference this year with special emphasis on the Regenerative Center. Hence, the name of our conference, taking the name, the same Mansoura University Regenerative Medicine Center, we organized our conference into two stages. The first stage here in Faculty of Medicine, 2 days, focusing on basic science, and the second phase in Alexandria in the form of satellite events targeting the different clinical application of stem cell. And this will be next 2 days. So this is our conference, the basic scientific evidence today and tomorrow, and satellite meeting will be in Alexandria the 2 days after this. And here I would like to express our thanks for all collaborators from different clinical branches who took the responsibility for supporting their clinical satellite events as a complementary part of our program. I would like to thank these professors with different specialties who are going to work in the second part of this conference. Now, a few photos about our institution. And these our students in one of the international meeting, they are presenting their paper and discussing in Holland. And this is the meeting by the Minister of Higher Education and our student. And happy this meeting also in Holland. And I would like to finish by this medical student. He was one of 2 medical student team presenting their work in Paris in the European Society of Nephrology and Renal Transplantation. They had 2 papers accepted, and this now we have fulfilled or achieved a dream to have undergraduate medical students having his paper accepted and is presenting it in a high prestigious international scientific meeting. And I will finish by that. Thank you.
بسم الله الرحمن الرحيم. وقل ربي يزيدني علما. والآن كلمة السيد الأستاذ الدكتور إيهاب سعد عميد كلية الطب.
بسم الله الرحمن الرحيم. الأستاذ الفاضل الأستاذ الدكتور سعيد عبد الخالق رئيس الجامعة، الأستاذ الدكتور مجد نصر نائب رئيس الجامعة للدراسات العليا، الأستاذ الدكتور محمد صبحي رئيس المؤتمر، السادة زملاء الحضور. إن التعليم الطبي يشهد تقدما ملحوظا في جميع أنحاء العالم في ضوء الفترات الهائلة التي حدثت في العديد من فروع العلوم الأساسية والتطبيقية، والتي تشكل في نهاية الأمر مخرجات من شأنها تخفيف الآلام عن المرضى وتحقيق الشفاء لهم. كل ذلك بالطبع بفضل الله أولا ثم بالمجهودات البشرية لتطوير الأداء الطبي. إن هذه المؤتمرات والندوات تؤدي إلى النظم العلاجية الصحيحة، والتي تشهد اليوم افتتاح المؤتمر العلمي السنوي لمركز البحوث الطبية والتجريبية بكلية طب المنصورة، وهي عن الطب التجددي. وهو الآن أتفضل بخالص الشكر للأخ الفاضل الأستاذ الدكتور محمد صبحي على مجهوده في مركز البحوث وعلى مشاركته الفعالة في كلية طب المنصورة ومشاركته في الأبحاث العلمية والدورات التدريبية في هذا المركز، وعلى مشاركته مع طلاب في إقامة هذه الأبحاث، والتي ساهمت في رفع شأن كلية طب المنصورة وكذلك جامعة المنصورة. وكما أوضح لسيادتكم كم كان جميلا من وزير التعليم العالي يوضح دور جامعة المنصورة وكلية طب المنصورة في تقدم التعليم الطبي للطلاب في هذه الحافلة بفضل هذه البرامج. كما تقدم بالتهنئة for Professor Elina John from the United Kingdom. Welcome in Mansoura, Professor Lee, because they do excellent effort to come at these days to present for our locality at this time. وكذلك أتقدم بالشكر لرؤساء الأقسام والسادة الزملاء الأساتذة الأعزاء كل في فرع تخصصه على مشاركته بناء كتيم وورك، والتي أتمنى أن يكون هذا هو العمل القائم لمصر جميعا. يتم الاجتماع على هدف شيء، ليس مهم من هو الرئيس، ولكن مهم أن تكون مصر في النهاية هي المهم لكي يتم الأمان لنا جميعا والنجاح للشعب المصري إن شاء الله. وأتمنى أن أخرج بتوصيات من هذا المؤتمر وفي تخفيف الآلام عن المرضى جميعا. ووفقهم الله جميعا لما فيه الخير والوطن الخير الوطني العزيز. انه نعمل نور ونعمل مصر. السلام عليكم ورحمه الله.
بنشكر السيد الاستاذ الدكتور ايهاب سعد عميد كليه الطب، والكلمه الان للسيده الاستاذه الدكتور ماجده نصر نائب رئيس الجامعه للدراسات العليا والبحوث.
بسم الله الرحمن الرحيم. أتقدم بالتحية للسيد الأستاذ الدكتور السيد أحمد عبد الخالق رئيس الجامعة، والسيد الأستاذ الدكتور إيهاب سعد عميد كلية الطب، والسادة الوكلاء، والسيد الأستاذ الدكتور رئيس المؤتمر الدكتور محمد صبح، وأتقدم بالتحية لجميع السادة الحاضرين. كما أتقدم بالتحية لبروفيسور لينا جونز. Welcome to the University of Welcome. حقيقة هذا المؤتمر له تميز من ضمن تميز الخاص بجامعة المنصورة وخاصة كلية الطب. فجامعة المنصورة تتميز بالعديد من المراكز الطبية، وشهرة جامعة المنصورة عالمية بأنها ذات التميز الطبي من الجامعات التي لها تميز طبي، وأحيانا يطلق عليها عاصمة طبية لمصر. من ضمن هذا التميز المراكز الطبية العديدة هذا المركز الذي يقوم بإعداد هذا المؤتمر اليوم، وهو مركز البحوث الطبية التدريبية الذي يرأسه الأستاذ الدكتور محمد صبحي. حقيقة هذا المركز بيقوم بيهتم بالجانب البحثي بجانب الجانب العلاجي والتدريبي وكافة المجالات التعليمية، ولذلك هو يتميز بهذا الجانب البحثي. هذا المركز كما ذكر الدكتور محمد صبح سوف يتم إعداد مركز آخر، مركز الطب التجددي، وهو يختص بأحدث المجالات العلمية البحثية. هذا المركز الذي يخص الستام سيلز الخلايا الجذعية بجانب مجالات أخرى، وهو مثل في المالتي ديسيبلينري أو المجالات المتعددة التي غير تخصصية في جانب واحد. فبجانبها بيستخدم علوم النانو والنانو تكنولوجي والعلاج الجيني. كل هذا سيتم من خلال تقديمه للريجنراتيف ميديسنز اللي هو المركز اللي سوف يتم في القريب العاجل افتتاحه مركز، وسينشأ عنه أيضا برامج علمية للدراسات العليا كما ذكر الدكتور محمد صبحي برنامج الماجستير برامج أخرى كثيرة في الطب التجددي. حقيقة يعني تميز هذا المركز أحس أن هو يحقق ما نقوله نظريا ما يخص البحث العلمي. فهذا المركز وطريقة إدارته من خلال مجموعات بحثية مفعلة بين كثير من الكليات، كلية طب وصيدلة وطب بيطري وكلية العلوم وكلية طب الأسنان. كل هذه الكليات بتشترك في أبحاث. البحث الواحد يشترك في العديد من هذه المجموعات، فهو يفعل بالفعل المجموعات البحثية. أيضا مشاركة الطلاب، ثقافة البحث العلمي للطلاب التي بدأت كما ذكر الدكتور محمد صبحي في المرحلة الأولى بمرحلة طلاب من كليات الطب والصيدلة وغيرها. بدأ أيضا تجربة زيادة هذه الثقافة في المرحلة الثانوية، وعلى علمي في مرحلة الطلاب بمرحلة الإعدادي. فهذا يعني له رؤية بعيدة في نشأ الجيل القادم على ثقافة البحث العلمي، لأهمية البحث العلمي بالنسبة لكل شيء من الناحية الاقتصادية ومن ناحية تطور البلد. الإحساس بأهمية البحث العلمي ويفعل هذا. أيضا أحس أن هو عملية الاشتراك وأهمية الفند اللازم للأبحاث ليس من ميزانية الدولة أو غيرها. فأيضا هذا المركز يشترك في العديد من المشروعات. مشروع على حد علمي ان هو في مشروع تم بالفعل اللي هو الSDGs ومشتركين في كثير من المشروعات، وهذه ايضا هذا المجال اللي احنا ندعو اليه البحث العلمي هو مشروعات توفير الجانب المالي من خلال مشروعات بحثيه او مشروعات كاباسيتي بيلدينج او غيرها للصرف على المشروعات. ايضا احس بتميز دي كليه الطب ومنها كما ذكر الدكتور محمد صبحي هذا المركز في خلال 2011 السنه الماضيه كان كليه الطب نشرت 85 بحث انترناشونال في مجلاتنا امباكت فاكتور، فدي نسبه عاليه نتمنى انها تزداد اكثر واكثر. اتمنى من كليه الطب وخاصه وايضا المراكز البحثيه ومركز الطب التكنولوجي ومركز البحوث التجريبيه ان يخوضوا مجالات تسجيل ابحاث ثقافه، نشر ثقافه تسجيل الابحاث كبراءات اختراع، لان هذه الثقافه يعني في جامعاتنا وفي مجتمعنا بتكون اقل يعني لسه ثقافه تسجيل ابحاثنا كبراءات الاختراع وكيفيه الاستفاده منها بعد تسجيلها، ليس تسجيل فقط، وكيف تحويلها الى يعني خروجها الى التطبيق فيما بعد. هذه الثقافه محتاجين ان هذا المركز انه يتبنى هذه الجزئيه كما تم خوضه في كثير من الامور خاصه بذلك. حقيقه نقطه امل ان ايضا كليه الطب ان تاخذها. كنا في زياره الى الصين ومن متقدمين جدا في اكثر من مجال في الطب والهندسه والزراعه، دول من اكثر المجالات تقدما، يعني تقدم جدا. يعني الطب الحقيقه لاحظت ان هو عصام كثير من البرامج. احنا يمكن بنسمع عن الطب الاكيوبانكشر اللي هو بالابر الصينيه وحاجات زي كده، احنا بننظر لها على انه مجال بعيد عننا. الحقيقه هذا الطب متقدم جدا عندهم، وهو يوجد هذا المجال في اللغه الانجليزيه ليست باللغه الصينيه فقط، واتكلمنا معاهم ان احنا ممكن نعمل حتى برامج مشتركه في هذه الجزئيه في التشاينيز ميديسنز. فاتمنى ان احنا ناخد هذا المجال لانه متقدمين جدا، ولمست هذا التقدم من امور بسيطه جدا. عدم التحنيط عندهم الحقيقه التحنيط قايمين هم عملوا اذا احنا نصور الزعيم اللي هو نامسون تاون ده وعاملين له تحنيط. انا تصورته تمثال شمع، ولكن هو يعني معناها ان هم وصلوا الى مراحل من التقدم في التحنيط. برضه العلاج الحقيقه يعني من بعض ما سمعنا ان هو ممكن اي واحد يكون عنده حاجه زي البرد والانفلونزا واحتقان ان احنا نقعد ناخد انتي حاجات كتير جدا، وفي خلال اسبوعين او ثلاثه نبتدي نحصل شفاء. ولكن لو حسوا انه من ذكر المستشار الثقافي واصابته ببرد وفي خلال يوم واحد بعلاج معين مشترك ما بين تشخيص يعني مجال الطب مع الصيدله في التقدم الحديث، فكان بيحصل شفاء شبه كامل. فارجو ان احنا نخوض هذه المجموعات البحثيه، يبقى ليس يعني زي ما احنا مثلا في نظام مانشستر ونظام التراديشنال، ارجو ان هو ان احنا برضه نخوض هذا المجال اللي هو التشاينيز ميديسن في هذا التقدم. مش عايزه اطيل على حضراتكم، بشكر حضراتكم جميعا، و يعني في النهايه اتمنى لحضراتكم التوفيق في هذا المؤتمر، وبشكر جميع القائمين على هذا المؤتمر.
الشكر كل الشكر.
كلمة السيد الأستاذ الدكتور رئيس الجامعة الدكتور السيد أحمد عبد الخالق في ختام الجلسة الافتتاحية. فلنتفضل سيادتك.
السلام عليكم ورحمة الله وبركاته. بسم الله الرحمن الرحيم. زملائي الأعزاء، أستاذ الدكتور إيهاب سعد عميد كلية الطب، أستاذ الدكتورة مجدة نصر نائب رئيس الجامعة للدراسات العليا والبحوث، الزميل الفاضل أستاذ الدكتور محمد صبحي مدير مركز البحوث والتجارب الطبية الطبية التجريبية في كلية الطب، السادة العملاء، السادة الوكلاء، زملائي الأعزاء من أعضاء هيئة التدريس والباحثات والباحثين، يسعدني أن أرحب بكم جميعا هذا الحفل العلمي المتميز. واسمحوا لي please allow me to pay a warm welcome to Professor Elena from Leeds, from the UK, the country which I love the most, the country where I have been graduated in Kiel University in Britain. So you are quite welcome to Egypt in general and to Mansoura University in particular. زملائي الأعزاء، يسعدني أن أقف أمامكم اليوم في هذا الحفل العلمي لأعبر عن سعادتي لأنه يوجد في جامعة المنصورة بؤر كثيرة مضيئة يأتي على رأسها هذا المركز المتميز الذي يديره أستاذ متميز يعمل في صبر وصمت. لم أسمع منه منذ أن كنت نائبا للدراسات العليا والبحوث مشكلة واحدة أو شكوى واحدة، وإنما في كل مرة يأتيني باقتراح جديد، يأتيني بإضافة، وحينما يدخل يقول أنا مش جاي لك في مشكلة، أنا جاي عايزين نعمل حاجة جديدة. فأنا باسمكم جميعا وباسم شخصيا أحيي الأستاذ الدكتور محمد صبحي على جهده المتميز. بأمانة، هذا الرجل الذي يعمل في صبر وصمت ويتسم بأخلاق العلماء بحق، والذي رفع رأسنا في جامعة القاهرة في المؤتمر الأول للأبحاث الطلابية الذي حضره معالي الوزير. وتقدم الطلاب من كلية الطب من جامعة المنصورة من مانشستر ومن غير مانشستر، وكنت سعيد للغاية بما رأيته من طلاب تدربوا وتمرسوا على كيفية إعداد البحوث وإلقاء البحوث وما يتمتعون به من ثقة غالية. وحصل طلاب المنصورة من كلية الطب على 4 مراكز أولى من بين 10 مراكز على مستوى الجامعات المصرية في الكلية. هؤلاء تخرجوا في طلاب في كلية الطب لسه حتى ما تخرجوش، ما بالنا حينما يتخرجون إن شاء الله. وهذا يعني أننا نملك القدرة، وأننا يلزم دائما وأبدا أن نتحلى بالثقة، ثقة بالله والثقة بالنفس. أننا نملك القدرة، نملك أن نكون جيدين، نملك حينما يكون لدينا هذه العقول الواعدة والمتميزة، نملك أن نكون متميزين. نملك في الجامعة أن نكون جامعة متميزة إذا ما اهتممنا بالبحث العلمي، ونملك أن نكون كما يقولون ويل كلاس يونيفرستي، نملك أن نكون ذلك. وفعلا أصبحنا في عداد الجامعات حينما احتلت الجامعة المرتبة 96 على مستوى أفضل 100 جامعة في العالم لم يمر عليها 50 عاما. الجامعات كده والهاير تايمز هاير education، الرانكينج اوف تايمز هاير education، ranking Mansoura University as the 96th university among the universities, the most developed universities, which its age about or under 50 years old. فهذا فخر عزيز. كيف تم؟ بفضل العلماء، بفضل الباحثين، بفضل العملية التعليمية في جامعة المنصورة. وهذا ما أعنيه أننا نملك أن نكون جيدين وممتازين. ولذا نشعر دائما في إدارة الجامعة أننا علينا مسؤولية كبيرة أن نساعد في هذا الخصوص، أن نمهد الطريق، أن نمد يد العون. ولذلك منذ 3 سنوات وقت أن كنت نائبا للدراسات العليا وبدأنا بالفعل ولأول مرة في إعداد خطة للدراسات العليا والبحوث في جامعة المنصورة، واعتمدت من مجلس الجامعة، فأصبح لدينا خطة متكاملة ل3 سنوات للبحث العلمي
الذي يمكن أن يتم دعمه من صندوق البحوث للجامعة. ونقدم الدعم للبحوث حوالي 2 مليون كل سنة في إطار إمكانياتنا وفي إطار ما نملك، ونتمنى المزيد. ندعم الإفادة الخارجي للزملاء يذهبون إلى المؤتمرات العلمية، ورفعنا حجم ما يتم دفعه للزملاء لكي يستطيعوا أن يحتكوا بالعالم. وكما قالت سيادة النائبة الآن، الاحتكاك يعطينا خبرات كثيرة. الزيارة الأخيرة في الصين لمدة أسبوع استفدنا في أمور كثيرة ربما يأتي الوقت لنتكلم عنها. ورفعنا قيمة ما يحصل عليه زميل إلى أقصى ما تحصل عليه أي جامعة مصرية، هو 8000 جنيه للزميل الفاضل الذي يذهب إلى هذه الجامعات. أيضا في إطار الممكن تم إعداد خطة ثلاثية لتحديث وتطوير المعامل على مستوى الجامعة كلها، وضع خطة لإنشاء المعامل المركزية في الجامعة. هناك اتجاهات عديدة في مجالات مختلفة لتطوير البحث العلمي، لأنني شخصيا أؤمن أنه هو الطريق للتقدم، وأن رهان الأمم على التقدم وعلى أن يكون لها موقع قدم أو موطئ قدم في هذا العالم هو لابد أن يكون من خلال البحث العلمي والتطوير. وإذا كانوا يقولون دائما أن الحرب في فترة السلام هي المنافسة، هناك سلام الآن فيش حروب، لكن هناك حرب تدور دائما لا تخمد، وهي المنافسة بين الأمم، وأن يكون لها السبق. المنافسة الآن هي منافسة مبنية على التقدم والتطوير الذي ينبني على البحث العلمي، ولا مجال لنا لذلك. وهنا تأتي عظمة قرآننا العظيم وعظمة الخالق سبحانه وتعالى حينما يحث على العلم والبحث العلمي والتطوير، ونحن بصدده الآن. لو ربطنا بين ما يحدث في الواقع وما نقرأه في القرآن وربما نمر عليه مرور الكرام، نجد أنه لا مجال لنا إلا بتطبيق كلام الله سبحانه وتعالى فعلا لا قولا، لا مجرد كلام نبحث. وربنا سبحانه وتعالى حينما يقول في أنفسكم أفلا تبصرون، نحن الآن اليوم بصدد ذلك في الطب التجددي أو الطب التجديدي كما يفضل أن يسميه البعض، Regenerative Medicine، طب تجديدي أو الطب التجديدي، سواء بالبحث عن خلايا جديدة أو أعضاء جديدة أو جلد جديد أو في كل ذلك لذلك لإعادة تنشيط الخلايا التي هلكت. وهذا ممكن أن يتم اوتوماتيكيا في جسم الإنسان كما تعلمون أكثر مني، أو ربما يتم في المعامل ثم يزرع في الإنسان. وأصبحنا نسمع في العالم كثيرا أن هناك قصبة هوائية زرعت في كذا في إيطاليا، وهناك عين تم تجديد عودة الإبصار مرة أخرى. كل هذا بفضل علم. أعتقد أننا في جامعة المنصورة نملك من الكوادر، نملك من الأساتذة، نملك من الباحثين كبارا وشبابا من هم لديهم القدرة على ذلك. السادة العلماء، هذا المؤتمر أحييكم عليه، لأنه مؤتمر لا يبحث فقط في زرع الخلايا أو الأعضاء أو إلى آخره، وإنما تبحثون في زرع الأمل، وما أجمله من أمل بأن نتمتع بصحة جيدة وبعون الله ربما عمر أطول. ونسأل الله سبحانه وتعالى لكم التوفيق، وجميعا نسأل اللهم احمي مصر من كل سوء، وكل عام وأنتم بخير، والسلام عليكم ورحمة الله وبركاته.
الجلسة الجاية بعد البريك إن شاء الله في قاعة أخرى جنبنا عن بعد 10 فدلوقتي نبتدي الكفر وشكرا.
دكتور فرحة معنا في direction chairperson for this third session. this third session to deal with the present and future of MERC. The present presentation will be done by Professor Ahmed Badawi, Professor of Gynecology and Obstetrics at Mansoura University, who will speak about his project of ECTDF and health sexuality.
Thank you, Mr. Chairman. Dear colleagues, and this talk, and I think in the coming few talks, we are going to show you some of the research projects running in the MERC. One of these projects about manufacturing new oocytes to treat patients with premature ovarian failure. The official name of the project is Experimental Manufacturing of New Oocytes, both in vivo and in vitro, to treat induced ovarian failure. It's one of the STDF. For my colleagues who don't know, the STDF is the Science and Technology for Development Fund. It's a competition, some sort of competition. You win £1 million to cover the projects. It's one of the £1 million projects. I'm the principal investigator, honored by the presence of Professor Muhammad Sobhi, Professor Fatma Husseini, Professor Muhammad Ahdi as the principal investigators, and many of my colleagues, Dr. Ahmed Lutfi, Dr. Muhammad Ahmed Sobhi, Dr. Sara, and a lot of administrative team working in the MERC. The idea of the project is as follows. To simplify the story, traditional teaching used to tell us that the female has got her own oocytes during the intrauterine life. At 20 weeks of gestation, the ovary of the female fetus contain about 5 million oocytes. And at the time of delivery, by the process of apoptosis and atresia, they are reduced to about 1 million follicles. When the female reaches the age of puberty, 350,000 follicles are present in each ovary. That's what traditional teaching is saying. And this is the cohort she is going to consume all through her reproductive life. When she finishes these follicles, she is going to be menopausal. When the follicles come to less than 1,000 follicles. Traditional teaching used to tell us that there is no renewal of the oocytes. When they finish, they finish. When they stop working, they stop working. But this is actually, in the real life, very unfair play, because always the oocytes are in danger. There are situations in which the oocytes are lost under the effect of alkylating agent, the chemotherapy. You should know that 10,000 female children per year undergo chemotherapy for example, non-Hodgkin lymphoma. And 80% of them are going to survive, but they are going to survive infertile because you have lots of zero oocytes in the ovary. The young patients below the 40, about 15,000 breast cancer patients per year below the age of 40, and more than 5,000 cancer cervix per year under the age of 40. They are treated with radiotherapy and chemotherapy. They are going to be cured, but they are going to stay infertile. This is not the style of life you'd like to see. Those young girls who are exposed to removal of the ovary, whether prophylactically or due to the presence of tumors, those who have got autoimmune problems, some autoimmune disease attack the ovary, causing lymphocytic infiltration of the ovary and destroy the cohort that we say that, according to the traditional teaching, is not renewed. There is 1% of the ladies are belonging to this group, familial premature ovarian failure. At the age of 15 or 20, they stop menstruation because they have finished the cohort. For all those girls, we don't have a solution according to the traditional teaching because there is no renewal of the oocytes. Recently, we developed a new group of patients we see as a worker in the reproductive medicine, those who marry late and they would like to have children. We have a group of patients who are called the poor responders, above the age of 40. They still have oocytes, but the oocytes are very poor to make children. And for this group, you need a fresh, new, young oocytes to make the job. This is a very big group of patients. Not that old, definitely, but some sort. Some sort. The current status of treatment is oocyte donation. It's called egg donation or egg sharing. That somebody is going to donate her oocyte or share her the oocyte, whether they donate or sell it. And you can see sort of this advertisement on the net that somebody is asking for earn $750 for egg donation. And you should know that one of the commonest sources of funding the education for girls in the United States is egg sharing and egg donation. But it gives her the oocytes, and you make from the sperm of the husband babies by the technology of eggsy, and put it in the uterus of that old female or that desperate lady, and then you have a child. But this is totally unreligious, and it's not accepted in many of the communities. So there should be a solution of it. The solution should come from new oocyte formation. Before starting the project, there is a good evidence to support the idea of new oocyte formation. That's why we designed this project. The objective of this project is to have a new oocyte formed, candidate for fertilization by the technology of eggsy and IVF in non-functioning ovary. So before designing the project, we had good evidence that this is possible. And I think on Thursday we are going to speak about the story of new oocyte formation and potential success in more detail. But I'm just going to tell you about the project. The project is going through 3 phases and is going to take the coming 2 years. The first phase is having the model. We have to have a premature ovarian failure model. So using the cyclophosphamide injection, we designed this model from the mice, and this is the first time actually to study the model in the mice using pathologic immunohistochemical studies, illustrating the acute and chronic effect, chronic damage which occur under the effect of chemotherapy. We have to have a model like what we have in the real life in the female. In phase 2, and I'm going to show you what we have achieved from this project. In phase 2, we will go into 2 directions. The first direction is in vivo stem cell therapy using transplantation of autologous bone marrow stem cell, adipose tissue stem cell, or peripheral blood stem cell. We will have a stem cell from these 3 sources. We are going to separate them, identify, propagate, and then we will use them in vivo for treatment of this damaged ovary in the previous model. And at the same time, one of the sources we can get oocytes, stem cell for oocyte formation, is the germinal epithelium of the ovary. Germinal epithelium is the surface epithelium on the surface of the ovary, outside the tunica albuginea, outside the capsule of the ovary. So we will try, one way, other way, is to culture this germ cell from the surface of the ovary by scrapping. We will scrap it and then capture it outside until we have an oocyte-like structure. Then we will use it for injection. The phase 3, after obtaining this oocyte-like structure, then we will inject it with the sperm to have at least plus possessed stage. That is the target of, or beyond if we can get it beyond that. So this is what we are expecting in the phase 3 of the project. We have finished already the phase 1 of the trial. We had the model. We got 50 mice, and we inject them with the chemotherapy. We know exactly what is the time of complete destruction of the primordial follicle in the model. And this is the first time for the description of a model of premature ovarian failure. We know how the repair is going to happen and what is the time. So the proper model has been obtained. It's a lot of work done on that. Now we are in the preparatory part of the phase 2. We are preparing the stem cell from the peripheral blood from adipose tissue, and we have completed the bone marrow already. We are preparing for the culture of the germinal endothelium from the ovary. So you see that it's actually a race for the formation of the new oocyte. There is achievements in this. You shouldn't stop at the stage of thinking about. You have to go rapidly, because if you cannot achieve it, other people are going to achieve it. Thank you very much.
I don't know if you wanted to postpone the question at the end of the session, or you wanted to take it now. Okay. 3 questions are allowed now. في حد يحب يسأل أسئلة من الشباب؟ Well, first we thank Professor Ahmed Badawi for this lecture of hope. شباب هيسأل لا شباب برضه اتفضل.
دكتور أحمد، بالنسبة للعنوان manufacturing يعني صعبة شوية، لكن ممكن نقول formation ولا ايه، ولا انا مش يعني manufacturing ايه؟ What's the meaning of manufacturing?
I hear you. This is the title which was sent to the SDDI. To win a competition, you have to be impressive. So the idea, because we learned from you that there are rules to win a competition like this. One of the golden rules become impressive to the judges, and the peculiar term sometimes is impressive, and it worked.
Yes. What about ovarian bank, like semen bank? In a female under the age of 40 can deposit the eggs in this bank for 20 years, 15 years, like semen bank. You know, we can store the semen for many years in young male who exposed to radiation or chemotherapy. What is about the ovarian bank?
I can answer this question well because I'm working in the field of IVF and aging, so I know well about the cryopreservation of embryos and of gametes. The cryopreservation of oocytes of sperms is absolutely successful up to 20 years.
Yes.
We don't have a problem. The oocyte cryopreservation is very difficult, and mostly we get the oocyte destroyed. So first is it's not a very successful policy. The second is obtaining the oocytes from young ladies like 16 years, 18 years, without being stimulated. We do it transvaginally using ultrasound scan, and this is again an obstacle, a very big obstacle. There are policies definitely of fertility preservation in cancer patients approved. One of them is to obtain oocyte, or to obtain oocyte and inject it with a sperm and preserve the embryo, which is more successful. But this is not working. The third is for whom you are going to do this, because sometimes there is a problem actually in expecting the event. Premature ovarian failure sometimes is unexpected event. Destruction of the ovary from autoimmune disease is unexpected event. The last point is there is a great debate about some of the malignancies have got cellular metastasis in the ovary, including tumors, neurological tumors. 25% they found the malignant cells in the ovary. So there is a question about having oocytes for a malignant patient, from a malignant patient, and preserve it and re-put it back into our body for fear of transferring again the malignant cell. So you still need the new oocytes definitely.
Thank you.
Other question?
Yes.
Microphone.
Yes. You are asking an excellent question, definitely, because usually people ask us.
What is the question?
What about the chromosomal number of the germinative cell? Yes, diploid number.
It's absolutely a nice question, and this, for the first instance, you think this is the impossibility of having a diploid cell is going to be a haploid cell. But this happened, and now it's achieved that meiosis is stopped, especially in the germinal epithelium. It's the easiest to start the meiosis is epithelial, germinal epithelium, because it has been programmed. We were speaking about the difference between undifferentiated mesenchymal stem cell and those who has got step forwards, like the stem cell in the ovary.
Have you actually induced meiosis in these cells?
We haven't yet, but it has been achieved. That's what I mean. There is reports about positive meiotic markers occurring. There is achievement actually.
Please present yourself and then ask the question.
أمل عبد السلام, Assistant Professor Anatomy and Embryology, Tanta Faculty of Medicine. I wanted to ask about the second stage, which is concerned with the mesenchymal cells in the ovarian manufacture immune oocytes. You had already make premature ovarian failure and then treat with the therapy. And I wanted to ask about what about in details about the therapy. This is the stage my questions about. Thank you.
Yes, you are asking about the core of the project, which I couldn't tell you, but let me tell you what is in the literature if you'd like to know. There is an achievement in the literature about that. Partially it has been achieved. But to be honest, it stopped at the stage of two-cell stage, and then degeneration occurred. The challenge is not in having the oocyte-like structure. The challenge is to continue to blastocyst stage, because this is the real start of the embryo.
دكتورة منال البرماوي, أستاذ مساعد أناتومي طب طاقة. You said that you will prepare oocyte-like structure. So we have some difference between the normal oocyte and the oocyte-like structure which will be produced by this experiment. Okay, so also the blastocyst will has abnormality. What is the abnormality which can expected in this blastocyst? What can we expect? The embryo has many abnormalities, I think, like this, or we have normal embryo.
Yeah, I got the point. You say an oocyte-like structure just to be complete. Nobody can swear that he's going to make an oocyte. The scientists usually call this oocyte-like structure until it forms the blastocyst. So what we are going to do when we have this structure, then we will do everything to be sure that it's going to, we will do the markers, we do the meiotic markers, we will grow it, then we will inject it with the sperm. We cannot, or the scientists always don't call it oocyte until it forms blastocyst. Because when you attend the further talks, inshallah, in Alexandria, then you would see that there is achievements at this stage, but they couldn't proceed into blastocyst. So you cannot call it oocyte until you form a blastocyst.
Okay, thank you. I think thank you very much. But I have a question. Could it be applied also not only for induced ovarian failure, but with primary ovarian failure?
Well, that's right. It's one of the group of the patients who are, of the models, of the patients who are the target is primary ovarian. So long as she has not the structure of the ovary. The first design of the project actually was a human. But we couldn't get funds from human therapy at the moment, so we are making a model.
Thank you very much. Okay, our next speaker will be the maestro of this work of MERC, Professor Mohammed Sobh, one of the eminent professor in Mansoura University or Egypt. He will kindly give a talk about also an STTF project. Professor Mohammed Sobh, please.
Our center was lucky to win 3 projects, STDF project dealing with the kidney. 2 of them, which I'm going to discuss today, dealing with a model of nephrotoxic nephritis. We focus on cisplatin, as cisplatin, as we know, is commonly used as anti-tumor agent and usually is followed by chronic renal failure. So we have 2 projects. The first one, this is the code number of this project, and this is the title: Comparison of stem cell derived from human amniotic fluid, human adipose tissue, and rat bone marrow for treatment of cisplatin-induced kidney disease in rat. This is the first project. The second project is to try to compare between different type of mode of injection of one type of stem cell, which is bone marrow derived mesenchymal stem cell, and its effect or its therapeutic effect on cisplatin-induced acute kidney injury or kidney injury in rat. Bone marrow derived mesenchymal stem cell for treatment of cisplatin-induced kidney disease in spread down the rat, study of impact of route of administration. Okay. First, we know that cisplatin is a heavy metal compound with a wide anti-neoplastic activity on a variety of solid malignancies. It cannot be replaced by the less toxic carbamazepine for treatment of many tumors, including germ cell tumor. So we are obliged to use this cisplatin. Cisplatin is mainly excreted by the kidney, so there is, of course, renal nephrotoxicity, and chronic and cumulative renal toxicity are the major limitation of its use. Nephroprotection must be maximum when cisplatin is used in a curative intent against germ cell tumors, since most of these young men are long-term survivors, so we have to keep their kidney. Although acute renal failure is now a rare event with the adoption of many strategies to protect the kidney, yet chronic alteration of kidney function are frequent. reduction in the glomerular filtration rate occurred in 20 to 30% of patients despite intensive prophylactic hydration and forced diuresis. Such change in renal function are essentially and unfortunately irreversible. So we have to tackle this problem, and in the project 1061, the aim of this research was to isolate and in vitro characterize the mesenchymal stem cell obtained from human amniotic fluid, human adipose tissue, and rat bone marrow. Then after that, we will compare and test in vivo the hypothesis that treatment with these cells could improve renal function and attenuate tubular damage in kidney disease induced in rat by injection of cisplatin. In the other project, so in the first project we are comparing different types of cells, stem cell. In the second project, the aim of this research was the same, is to isolate and in vitro characterize other bone marrow mesenchymal stem cell. After that, we'll test in vivo the hypothesis that the treatment with mesenchymal stem cell of other bone marrow origin could improve renal function and attenuate tubular injury and kidney disease induced by Sprague-Dowley rats by injection of cisplatin. In addition, and this is the main difference, we will study the impact of different routes of administration. Either we'll inject it peripherally in tail vein or directly into the renal artery or directly into the kidney tissue. So we'll compare the route of administration and its effect on the therapeutic potential of these cells. This is a busy slide, but I will describe it. We have here 6 groups. Group 1 is the first group in which rats were given cisplatin. This was injected intraperitoneally in a dose of 7 milligram per kilogram, by the way, in 1 milliliter saline. So this is the positive control group. The second group, in which cisplatin was given, and 1 day after we gave bone marrow derived mesenchymal stem cell, rat bone marrow. The third group, cisplatin was given, and 1 day later, the animal was treated by stem cell driven from human amniotic fluid. The fourth group, cisplatin was given, and rat were treated with stem cell derived from human adipose derived stem cell. So these are the treated group, group 2 and 3 and 4, with different type of stem cell. Group 5, we gave cisplatin and we treated animal by culture media. And the last group is normal rat treated, just injected by saline. So this is negative control group. So we have negative control, normal, positive control, diseased, untreated, and other 3 groups treated either by bone marrow, derived, adipose, or amniotic fluid. And we have another group, we can say control treated just by media. The second project, the same group 1 was cisplatin treated, diseased animal, and group 2 was treated by bone marrow derived stem cell, mesenchymal stem cell. Either in group 2A, the drug was injected into the left renal artery. 2B, the drug was injected into the tail vein, and 2C, it was given in the tissue. So different mode of administration. Group 3, we gave culture media by the same way, and group 4, negative control. So we have, this is the design of the 2 experiments, of the 2 protocols. Now, both animals in these trials were subjected to culture work on the stem cell, how we extracted stem cell from bone marrow, from amniotic fluid, from adipose tissue. This technique will be described by my colleague Muhammad now, after a while. And also, the stem cell were subjected to in vitro study. We look for fibroblast-like colony forming unit. This is stem cell, and also in vitro differentiation to assure the stemness of these cells by testing the osteogenesis assay, adipogenesis assay, chondrogenesis assay. So now we are sure we have stem cell ready for injection. Also, these cells were subjected to flow cytometry and real-time PCR. I think Muhammad will come now and show us how we injected these stem cells and how we extracted them.
Isolation, purification, culture, and decarbonization of mesenchymal stem cell derived from different sources. Number one source, rat bone marrow, human adipose tissue, human amniotic fluid. Rat bone marrow, adult inbred split dowel rats that weigh 180 to 210 grams served as bone marrow donor. Bone marrow has obtained was obtained from long bones of adult SCD rats for each experimental rat. Bone marrow cells were isolated from 4 donor rats. The bones were sliced by immersion in 70% ethanol. The ends of the bones were cut, and the bone marrow was extruded by inserting a needle in the one end through the bone shape, and the injection of tissue culture media TMAM containing 10% hydrogen serum. The effluent was collected in the sterile tube. Gentle pipetting resulted in the obtaining of a single cell suspension. Second source, human adipose tissue. To date, we have obtained stem cell from virtually all over the body in the hopes of finding ways to avoid the ethical issues associated with the embryonic stem cell. Newer research studies have now looked at the potential to use stem cell sources from fat. The benefit is that fat is readily available and can be extracted without complication compared to some of the other methods of stem cell sourcing. Excess of adipose tissue fat is often available in the form of restricted tissue from elective plastic surgery, procedures lipotomy, for example, abdominoplasty or breast reduction. Lipoaspirate from liposuction procedure is also a convenient source of tissue for isolation of human mesenchymal stem cell. The fat tissue is lipoaspirate is already significantly associated, so processing is simplified. The tissue was placed in a sterile specimen container at the time of the harvest. Tissue was stored at ambient temperature and processed within 8-hour refrigeration was avoided if possible because the lipid hardened at low temperature. Tissue may be stored overnight at 4 degrees centigrade, however, it should be chlorinated at ambient temperature prior to processing. 50 grams of adipose tissue or 50 ml of lipoaspirate was conventionally processed using 15 ml sterile disposable centrifuge tube. A number of select sources of human amniotic fluid, multipotent stem cells are also found in amniotic fluid. These stem cells are very active, expanding extensively without failure and are not chlorogenic. Amniotic stem cells are multipotent and they can be differentiated in cells of adipogenic, osteogenic, myogenic, endothelial, hepatic, and also neural lines. Use of stem cell from amniotic fluid overcome the ethical objection to use human embryos as a source of the cells. Here, the baby swimming in amniotic fluid cell placenta, mesenchymal stem cell was isolated from amniotic fluid of pregnant women who underwent a cesarean delivery for breech presentation. The samples were collected by penetrating the membranes after the uterine muscle was opened for the cesarean delivery. Cells were isolated from the amniotic fluid sample no more than 4 hours prior to use. Here we make in vitro fibroblast-like colony forming good assay. Mesenchymal stem cell, after being isolation, characterized by their adherence to plastic coincidence of the heterogeneous cell population with a prominent sphenic shape morphology and were able to form colony-like shape. Here, and primary culture of rat stem cell stem cell aggregate in colony for in rat mesenchymal stem cell. Also in the human adipose tissue, cells aggregate in colony like 3 colonies here. Big colony from hematocrit fluid, we see a big colony from mesenchymal stem cell in primary culture. Cells were purified in culture media with different batches. The mesenchymal stem cells were generated by standard procedures and grown for at least 2 batches in culture. Contaminated hematopoietic cells were depleted during batch 1, and the mesenchymal stem cells were morphologically defined by a fibroblast-like appearance. Here, in the batch 3, rat bone marrow mesenchymal stem cell, like carpet conference shape. Also, another slide, rat bone marrow, spindle shape, very well. Human adipose tissue with spindle shape in batch 3. Here, human adipose tissue also fibroblast-like in batch 3. Culture here, hemolytic fluid stem cell at batch 3. Here, another slide for hemolytic fluid stem cell. In vitro differentiation, I say osteogenesis. For that, after culture of mesenchymal stem cell with osteogenic induction medium for 14 days, most of the cells showed the aggregates or nodules of calcium mineralization in the culture by alizanide staining. Here, cells aggregates in the alizanide stained with alizanide red, forming nodules from calcium minerals. Also, aggregate cells stained with alizanide red, forming minerals of nodules of calcium. Here, human adipose tissue differentiated to osteogenic. There is nodules from calcium. Also, stained human hemolytic fluid stained with alizanide red. In vitro differentiation is say adipogenic. Adipogenic differentiation was apparent after 1 week of incubation with adjuvant induction medium between the culture period of 3 plus or minus 2 weeks. About almost all cells obtain numerous oil red or positive lipid droplets. Lipid droplets stained with oil red from rat bone marrow. Also another slide, oil droplets stained with oil red from rat bone marrow. Here also human adipose tissue stained with oil red, droplets stained with oil red. Cells from human adipose tissue. Here another slide, new slide from hemolytic fluid stained with oil red. Here also stained with oil red. In flow cytometry for surface expression, say the analysis revealed that the expression of surface antigen of mesenchymal stem cell derived from human adipose tissue were negative for CD14 and CD34, while they were positive for CD29, CD90, and CD105 and CD13. The slide shows the positive and negative flow cytometry of the cells obtained from human adipose tissue. The analysis revealed that the expression of surface antigen of rat mesenchymal stem cell were negative for CD45, while they were positive for CD29 and CD90. Here is a rat mesenchymal stem cell flow cytometry slides. Thank you. Now I will present. Yes. Another project we have dealing with different route of administration, different administration stem cell. Here, first type is stem cell injection through tail vein. Here, anesthesia or elimination rats inside this. In our MERC operation room, we operate this injection. Here, rats immerse their tail in hot water for phosphorylation. Try to inject the tail in the tail vein. Here, the rat is sleeping for continuous smelling hallucin from this tube. And after injection, we close the point of injection by cotton. Injection of stem cell under renal capsule. Here should be shaving here and sleeping the rats in the one side, and strike the side of the transplant by ethanol and betadine. Here try to scratching the membrane of renal capsule by syringe with stem cell and inject under renal capsule for forming capsule. Here I'm trying to inject After that, you can use the third type by using surgical before and after branching by clamping all block before and after renal artery. Here and try to insert the needle 25 G. Here I inject now. Now I will show some movie for this injection, like the tailback injection. You can say insert the syringe, slowly injection the tail vein. Another movie for operation of renal capsule injection. Now I try to scratch the surface of the membrane by fine scissors for make poor protection. Insert a small needle of cannula under the membrane for making capsule. Just for clean. Here you can see this balloon forming here. After that, increasing the size by inject another volume. Then inject the stem cell with the media under the capsule here. You can see the needle range. After that, I close by skin. Another movie for renal artery injection. This is an artery. Here I clamp it by the bulldog. two clump is here shown for renal, for aorta, mid aorta, before and after renal artery. I try to show the side of renal artery. Here, the set is a mesentery or pads around the artery. It's very fine needle. It can under the microscope to show as bigger. Now inject stem cell. The last movie for showing the isolation of stem cell from bone marrow. Cutting the hind limb of rats. Then I separate the hind limb. Separate for femur and tibia fibula. I remove the muscle from around the bone. Then I present four separate tibia fibula and the femur and put it in a sterile tube. Transfer after that to tissue culture back in laminal flow. For first point is immersion in ethanol for sterility. After that, cutting the head of each bone for inserting needle. After that, insert needle inside each bone separately for flushing stem cell in the another clean tube. That I collect all stem cell in one flask, then incubate in culture media. That's a primary culture for stem cell from rat bone marrow. Thank you.
Now I'm going to present the result. We have results. Okay. So we have two protocols. One to compare different types of stem cell and its impact on cisplatin-induced kidney injury, and the other protocol in which we use bone marrow derived mesenchymal stem cell and injected by different route of administration, as Muhammad showed us. So this is the chemistry. Serum creatinine in using different types of stem cell. And you see here the yellow. Okay, this is a normal. This is a normal creatinine. You remember a control negative group. Normal data. This is a normal group, normal creatinine. And this is a group of animal treated with given cisplatin. We show there is rising in serum creatinine, like that. You have two colors. This is the cisplatin untreated, and the cisplatin treated with just culture media. The same, no effect. Significant difference when compared with the negative control. And this is the animals treated by two types of stem cell. The bone marrow derived, you see significant difference here, and the adipose tissue derived mesenchymal stem cell. Of course, this is a project is still working. We finished half of our work, so we still have to use amniotic fluid derived. We did some experiment, but not yet fully finished. And also we still have some work with the adipose drive. But kidney function, there is significant improvement in kidney function using these two types of stem cell when compared with the untreated group. If we look to blood urea nitrogen, the same. This is the normal rat, BUN. This is the cisplatin treated, untreated, and cisplatin treated with just culture media, significantly high. And this is the other two groups, bone marrow derived and adipose derived. So from the chemical point of view, we showed significant improvement on treating with these two types of stem cell. We look for the oxygen-free radical superoxide dismutase. This is the normal. This is the untreated and treated with the culture media, and this is the treated with two types of stem cell, significantly better than those untreated. So it seems action on oxygen stress is one of the mechanisms by which stem cell work. Besides, of course, the possibility of trans-differentiation. This is the histopathology. This is the group 1. Cisplatin treated rats. You see, this is the outer medulla, this is the cortex, this is the medulla, this is the outer strip, outer medulla. You see the damage in the renal tubule. So there is extensive damage or necrosis in the tubular cells. And here we compare at the same dawn after 4 days. This is the stem cell treated group, and this is the untreated group. You see the extensive tubular necrosis and damage. This is untreated group, and this is the stem cell treated group. You see, this is the tubular line. If you compare, there is intent to heal, regeneration, and you see these solid sheets of cells. It seems these are the stem cells coming from blue, trying to do regeneration. At 4 days, there is significant histologically better than in treated than untreated group. This is the same animal, the same study in which this is the untreated. You see tubular damage, necrosis after 7 days, and you see how the regeneration is better at 7 days. And also, this is this is the cisplatin injected, and this is 11 days after sacrifice. Of course, this animal is to be followed up for up to 60 days, as we said, and we are in the halfway. But the data available shows histological and biochemical improvement when we treated animal by different types of stem cell. Now, if we compare the different route of administration, is the same. This is the normal control. This is the untreated and those treated with culture media, and we have two routes of administration here. We have the tail vein and the renal artery. you see both of them gave favorable response, and you can find any significant difference between the two routes. So up till now, two routes of injection, local one and systemic one, in our model gave similar functional results. This is the PON, the same results, and this is the oxygen free radical, the renal superoxide dismutase, the same. The treated animal were significantly better than those untreated. This table is very busy, just to tell you or to show you how the posologic evaluation of our case, our experiment is very tedious. You see, this we look for the necrotic tubule and we give scoring. We count number of necrotic tubule per high power field, and we give scoring from 0 here up to 7. And we look for the atrophic tubule, we look for the mitotic figures, we look for the regeneration, and you see it is very extensive, and we do statistical analysis and we do correlation, and now we are halfway. We didn't finish our statistical analysis, but at this time point and within the available data, we can somewhat safely say that stem cells have therapeutic potential on cisplatin-induced kidney injury. Therapeutic effect of tested types of cells is comparable. Different route of administration up till now are therapeutically comparable. Besides trans-differentiation, a favorable action on oxygen-free radical is a possible mechanism to achieve improvement. We expect more interesting data to come out of this still running two research protocol, and thank you.
Now we can start some questions. I would like to start by myself. Have you any information about the use of special local applicability of renal tissue scaffolding niche loaded by the stem cell as a way of stem cell administration?
Renal-wise, I think this is used in other areas such as, of course, bone, cartilage, even in the heart has been matrix used. But renal-wise, all what I'm aware is that we give, as I said to you, as I said, peripheral or directly, but scaffolding in the kidney, possibly there are, but I'm not aware. In this area, we can use other, of course, scaffold is used to improve the action of the stem cell in treating the injury. But of course we have ideas, and I think we have protocol like this, to use damaged renal tissue, the extract of damaged renal tissue in the culture media, so that we know that when there is tissue damage, there is a release of certain cytokines which are important for the growth and differentiation of these stem cells. So we are thinking, I think we will do this. We have this protocol for that, in which we will use extract from tissue damage by the same disease model to compare it with ordinary culture media. But scaffolding, I'm not aware. To put it in the culture media, possibly it will show better therapeutic effect than if we rely on the conventional media. Just we are going to examine this possibility.
Any other question?
How the mesenchymal stem cells injected via red vein make homing to the renal tissue and make the same beneficial effect obtained by intrarenal injection? Is that via cluster designation, certain cluster designation, or what?
based on flow cytometry.
The phenomena, as you see, it is clear. We injected in the tail and in the artery, and we saw that local injection will give better result. But what we found is here, as you saw, there was no significant difference between the two methods we used. This could be explained by the cytokine released from the injured side. This cytokine will migrate through peripheral blood, and possibly these cells will follow this signal of cytokine and will do what's called homing. It seems that this cytokine is intense and the stimulus is too strong, so that a good amount of the stem cell will go to the proper side.
I wanted to talk about the first part about morphology of the stem cell. Morphology. When you extract stem cells and then you in vitro culture, you distinguish between different types just morphologically, right? Not only morphology.
We know that now we have bone marrow derived this in certain groups, and this is adipose derived. We know it.
But I speak about the percent of mesenchymal stem cells in bone marrow. Like you extract bone marrow and you culture bone marrow, and from first step you said it's stem cell. I don't think all of them are not stem cells. Some of them. So yeah. So and I think you distinguish by morphology. So I think it's better to show some morphological marker. Like for example, you state something specific to mesenchymal stem cells and shows the percent how many cells are mesenchymal from the whole culture. And if you can propagate, or because you need large number when you transplant, sure. But because this will give you like false number when you inject, you think that this is large number of stem cells, but sometimes it's not true. Maybe half, or we don't know.
Possibly through magnetic beads. Possibly, we can't say that all the cells you injected are 100% stem. And according to the graph which was presented by Mohammed, still you have some 4% of cells are not. But with the magnetic bead, in which you have the receptor and the antigen and antibody for receptor, so you can exactly 100%, these are the so and so. And of course when we go, we use the magnetic beads in some of our trials.
يمكن بس هو يمكن مختلط عليه الامر انه not only the morphology, but when you look and you see the spindle shape, you know that you are right, on the right way. But this is one of the criteria, not all the criteria of having mesenchymal stem cell. Please.
I think what you said is a very fair question in terms of purity of your cultures. Unfortunately, there's no marker for MSC that are applicable to every tissue. For example, adipose tissue are CD34 positive, whereas bone marrow 34 negative. And there's other markers, and there's still a lot of controversy in the field and people arguing. So I take your point, but it's very, very difficult at the moment to say that this culture is pure or not very pure. So it's work in progress, I would say. So maybe molecular markers would be better than surface markers, because surface markers sometimes not very useful. From the photographs, it looked like amniotic fluid cultures were more heterogeneous because there were more round cells and cuboid cells. So it needs to be taken into account. Yeah.
avoid this problem, I think it is a better way, and I'm going to do in our project, to have isolated bioinformatics to be certain specific cells or certain number, and to be sure this is, for example, this is mesenchymal stem cell, for example, 278 marker, to inject 1 million or 5 million or something like that. And I think this is a better way to save calcium.
Any other question? Thank you.